welcome you all to the Fall 2024 Applied Ecology Minors Research Symposium. Really quick, I want to celebrate that as of December, not 2023 last year, I should have updated the slide. As of December 2024, we'll have 167 students who have graduated from our program since its inception. Pretty awesome. Super, super proud and very glad that I have this auto-populating Excel spreadsheet that shows us all how we've grown over time. Before we get started, I want to acknowledge the lands within and surrounding present-day Raleigh are the traditional homelands and gathering places of many indigenous peoples, including 8 federally and state-recognized tribes: the Kawari, Eastern Band of Cherokee, Halua Saponi, Lumbee, Meherrin, Occaneechi Band of the Saponi Nation, Sapani, and Waccamaw Siouan. We share an ongoing responsibility to safeguard these lands and to respect the sovereignty of the tribes and indigenous nations residing in North Carolina. Our goals in applied ecology are to study the world around us and to apply what we learn to better manage or conserve natural resources. But I think it's important to remember that we humans are inherently part of nature, not separate from it, and that way before settler scientists decided this was important, expert knowledge and care for the natural world was already applied as an integral part of indigenous cultures. Acknowledgement is a form of decolonization, dismantling an existing power structure. In order to change systems of oppression, we need to first acknowledge that the system is built on oppression. North Carolina State University is a land-grant university, which means it originally received funding through the Morrill Act, federal legislation that bought or forcibly stole land from indigenous tribes. The sale of indigenous lands raised $134,920 to fund NC State. That's the equivalent of $3,810,578 today. I hope we can disrupt and dismantle colonialism beyond a simple territory acknowledgement by supporting and empowering each of our students as scientists. For this first session of our 3-part symposia, we're celebrating the research performed and completed by 4 different undergrad students. So with that, I'm going to introduce to you Trisha Belus.
Hi, my name is Trisha Belus. I'm going to be presenting on the research I helped conduct in the Sustainable Health Ecology Lab, with a focus on the immune response of water snakes to hemoparasites. Okay, so animals, including humans, are all exposed to parasites, either through water, food source, social contact, vectors, or the environment. In humans, some vector-borne parasites that cause malaria and Chagas disease, as shown. In animals, they have immune systems that help to reduce the damage so that the animal can continue to survive and reproduce despite of these infections. Today, I'm going to be specializing on these hemoparasites. So your immune system does most of the work in the blood. This is just a diagram in the middle of a blood smear from a snake, a water snake. Our blue cells are the red blood cells, the erythrocytes that carry oxygen throughout your body, and the white blood cells, specifically that pink one, is the heterophil, one of the leukocytes that you'll find in snakes, associated with the immune system of organisms. So some white blood cells are part of the innate immune response, as you can see on the left. Something to note is neutrophils are the associative of heterophils. Neutrophils are just in mammals, as are seen on this diagram. Heterophils are one of the most common white blood cells, and they are phagocytosing cells, which means that they can broadly recognize and engulf any foreign agents, such as bad bacteria. And then on the right, vertebrates also have an adaptive immune system that involves your lymphocytes. So your lymphocytes are cells that can learn to recognize specific antigens from pathogens that the organism has encountered previously. Reptiles do have the adaptive immune system, but it's not as robust and it's more limited than that of mammals. So if we're looking at the blood and we are counting specifically heterophils and lymphocytes, we can get a heterophil to lymphocyte ratio This will give us more information on how the two arms of your immune system are working. So we expect the ratio to be larger if we are, if there's a lot of heterophils in the HL ratio. So this would happen when the animal is injured and there is a wound and entry point for bacteria to enter, and otherwise other causes of high inflammation. This would also be high when the animal or organism is under high stress. So we expect the ratio to be lower if there are larger amount of lymphocytes. We would expect this when the animal is actively infected by a pathogen and is mounting that adaptive immune response. Other studies have shown that a lower HL ratio can be associated with pathogens for mammals and birds. But for this study, I'm going to be focusing on this relationship for water snakes. Okay, so specifically, I hypothesize that snakes infected by blood parasites will mount an adaptive immune response, and thus those that are affected by the blood parasite will have a lower HL ratio than those that are unaffected. So here we'll start with our sampling sites. Here are our field locations on that map on the left. And then on the right, you'll see in our physical examination slide, in the next slide, there's 4 species of interest. So from top to bottom are northern banded, plain-bellied, and brown water snakes. But something to note is that within our field studies, the ones that were captured and taken blood samples from are our northern and our plain-bellied. So that's first and third snake on the slide. Okay, so then once a water snake was captured, a physical examination took place. Any data was documented on this field data sheet, but I will focus more on the blood collection part. So each individual snake blood was taken near the vent, and then the blood was put into the small centrifuge tube. A capillary tube was taken, any of the excess blood to create a blood smear, having that feathered edge and a monolayer being the most important for our cell count protocol later. And then we did a right Jesma slide staining. So this will just help us differentiate the different types of different types of cells, which is really important when we're getting to our HL ratio. And then our cell count protocol. So our protocol was adapted by an article, Diagnostic Hematology of Reptiles, adapted by our postdoc, Kenzie Pereira. And to start, we started our slide at the corner, focus up to a 40x objective, and then moved in this up and down motion, taking note of any of the leukocytes until we had a combined total of at least 100. And this is just an example of some of the fields of view that you might see. So all of these slides that I'm talking about are ones that I have personally counted and used the same consistent method, but we're working on a project with multiple people identifying and counting white blood cells and also hundreds of blood smears. So it's very important that we are working out ways to ensure consistency across researchers, making sure that we are, for example, kind of ruling out slides or fields of view that are not great to be counting from. So for example, that smear on the top left is poor staining. There's not really a super distinct pattern that you'll see. And we want to make sure that the red blood cells and leukocytes and differentials can be easily identified. And then on the bottom, between both of them, there is a lot of clumping. We want to make sure that there's a monolayer that is easily identified. So making sure that between observers, we are ruling out those that are not good for counting. And this also might be that we're going to be counting the same field of view, which is those grids on the right. So this might be a way that we're implementing that we are going to be counting exactly the same fields of view and being consistent. Okay, so then parasite identification. 2 main blood parasites for this research. The top 2 is Serpentoplasma. The bottom 2 are Hepatozoon. So Serpentoplasma is most notably your circular, very uniform bubble inclusions in the cytoplasm of red blood cells, while your hepatozoon are those they look like rice grain. They're more ovular and elongated, usually also in your red blood cells. Something to note while we were identifying parasites is that their serpentoplasma infection is only suspected because there is limitations on certain bubble artifacts in the slides that may skew our identification of serpentoplasma. So that's just something to note. Okay, so then when we are graphing our total of 32 snakes, there were 5 with that hepatozoon infected. Positive infection is that 1 on our graph, and 0 is those that were not infected. It was found to have a significant negative relationship with that HL ratio, which is consistent with my predictions that the snakes will be mounting that adaptive immune response in response to the hepatozoon infection. So then out of those 32 snakes, there was 10 with suspected infection of serpentoplasma. This was not a significant negative relationship. There is, although it's still insignificant, it seems almost to be a trend with higher ratios for this. So this would not be consistent with my prediction that the snakes will mount your adaptive immune response and have a lower HL ratio, which brings into the fact that these are only suspected infections. This might introduce noise into the data set if we're saying that all of these infections were not, in fact, true, and we can't really rely on this data until we get more genetic testing confirm that the DNA is present for those infections. So going back to the prediction that snakes with blood parasite infections will have a lower HL ratio, this was confirmed based off of hepatozoon infections. This would suggest that HL ratio could be an indicator of pathogen infections, at least for that hepatozoon parasite. And then some of the applications is HL ratios could have several relevant applications for snake health. So either monitoring indicators of stress levels or pathogen infections within zoos or rehabilitation centers when snakes are in captivity, or overall HL can preemptively monitor snakes' health and allow for treatment intervention before the parasite levels could reach critical levels and then negatively impact the snake's health. And then for conservation initiatives, insights on snake's health and then subsequent stress can assist in breeding programs or immune response to relocation or other habitat management techniques. And then for future research, we talked about HL ratios and blood parasites, but we're also working on a larger project related to HL ratios and other indicators of snake immunity to emerging fungal disease, as you'll see on the right side of this diagram. We're looking at fungal disease load. So qPCR, which is essentially your concentration of fungal DNA found in the individual, the amount of lesions or scale abnormalities or the severity of those, and also the recapture probability, which may be an indicator of survival of wild snakes. So my research is contributing to that project, and I will also continue to be working on it in the future and next semester. So I'd like to acknowledge everybody in the Shell lab, including my wonderful people that I have in the first 2 rows. Especially Skylar for letting me participate and being my mentor. And also our postdoc, you'll see on the right, Kenzie, for letting me tag along on her project. Everybody has been very welcoming and very helpful in collecting data and creating this project. And so with that, I have just, if anybody would have any questions on my presentation. Okay.
So you said there were elevated levels, HL ratios in snakes that were suspected to have serpentoplasma,
Mm-hmm.
right? And that made you suspect that so you're checking with DNA to make sure, like, is it actually there, or were those air bubbles, right? Or
Mhm.
some other artifact. I'm curious, like, is there another situation that might explain elevated HL ratios for some parasitic infections, like maybe a new exposure to like a novel parasite that, you know, the immune system isn't used to, so it's still reacting to it?
Mm, that is a good idea. I definitely think within literature, serpentoplasma is fairly common in snakes. So I would think that they would have that adaptive immune response. Um, just because within wild populations, it's already kind of been there and reported there, but it is something to look at for sure. I'd be interested to see how the DNA turns out for them for sure. Yeah.
Consistent is the size of those parasites in the red blood cells.
So
Just based on the, yeah, some of those, we're looking at the little white circles inside the,
yes, so talking about like the serpentoplasma.
so some of them are kind of bigger than others, right?
Yeah, so I think it was mostly within, especially on the top left, it was regardless of if it was consistent, it was very prevalent. So it led me to believe that some of them might not be just artifacts just because they were so consistent in their placement and throughout the red blood cells on the slide. But there are some, like even on the right, you can see that there's really not as many. So it really just comes down to what the DNA would tell us. Yep.
We have, quantification of the HL ratio, but it also looks like you could have a quantification of the extent of the infection.
Mm.
Could that also be a factor to explain this? Because it looks like you had either infected or not infected.
Mm-hmm.
You quantify the level of infection. Could that help clarify?
Gotcha. So I think that could be a good hypothesis to see if the level of infection or level of cells that seem to be infected within a field of view, could that relate to the severity of your HL ratio changing? I think that that would be a good idea because even within, I did see a couple slides that had hepatozoon infection, but not as much as like your bottom left. I believe there's like 3 cells just within that snapshot that have that infection. There was a snake in particular that I really only found like 2 infected cells within all of the fields of view. So it could be viable that those with less infection, or maybe it had just begun and it wasn't as prevalent, that it could have less impact on the HL ratio. Yeah.
Are you excited to count all those infected cells?
I'm
You're just like, oh no.
I am so excited. I'm so excited. I'm ready. My next semester is open for counting cells.
Up next, we have Nicolas Galvez.
Hello, I am Nicolas Galvez. I'm a student here at NC State, minoring in entomology, but I also work at the Science Museum downtown on the 3rd floor in a very long named lab, but we're more commonly known as the Ant Lab. We have a YouTube channel and we focus on insect behavior and movement, specifically very high speed movement that most, that your eyes wouldn't be able to see. That does not work. Okay. And my research is on cockroaches. So cockroaches have been long known to be model systems for animal study but especially insect research. We know just about everything to do with cockroaches. We know how small of a gap they can fit through. We know the ins and outs of their legs and how they're built. But in 2011, they published a paper about a new species of roach in South Africa that, to me, looks just like a grasshopper, but is a cockroach, and it jumps for over 70% of its land locomotion. And in the introduction, I noticed that there was this sentence that said that no previously described extant species of cockroach jumps, which, as anybody who lives in North Carolina and has gone downstairs to their kitchen, you know, at night, cockroaches can definitely jump, and they're very good at it. So I set out to research this. I set out to describe jumping in 2 Paraplaneta species of cockroach, Paraplaneta fuliginosa, the smoky brown roach, and Paraplaneta americana, seen here, the American cockroach. I also set out to experimentally test the contribution of their hind legs in the jump, which we will get to. And I also wanted to determine if their behavior changes when crossing a gap or in different situations. And this is how I did that. So at our lab, we have very expensive cameras that I fear for my career if I dropped one. But I would set a roach on this platform here, and it's covered in a non-stick coating, so the roach can't climb down. Meanwhile, the camera is running, so it's continuously capturing footage of whatever's in front of it. And when I'm ready to save the footage, I would turn on this very bright light that shone directly onto the roach. Not only was the light necessary for the camera to capture visible video because it's shooting at 6,000 frames a second, but the light also encouraged the roach to move and try and find somewhere darker that was not, you know, under the full intensity of the sun, or it seemed like. So in this next slide, this is an American cockroach jumping off of the platform. Nope. Like access to the microphone. Okay, sure. So this is an adult female American cockroach jumping off of the platform at 6,000 frames a second. I can replay it. Nope, that didn't replay. There we go. Yeah. And I found that they do a very distinct series of movements. I labeled the start of the jump as when their body began moving forward. They very clearly knew that they were going to jump before this, but this was when I labeled as their jump starting. And I labeled the end of the jump as when the last part of their body left the platform. So they were no longer in contact with the ground. And on average for American roaches, it took around 35 milliseconds, so like a 30th of a second from start to finish. After they started their jump, they were pushing off with their middle legs and moving their hind legs forward until they locked them in place. So it took around 15 milliseconds until they locked their legs in place. And then once their hind legs were locked in place, their hind legs provided the majority of the thrust to the jump until their middle legs left the ground, and then they were completely off of the ground. However, there was a problem. These roaches had been living in a box their entire lives. We got them from another lab at NC State that does more roach research. And so this jumping behavior might not be indicative of a wild population. You know, maybe wild populations jump differently because they have free range. They haven't lived in a box their whole life. So to do that, I set out to catch wild roaches, smoky brown roaches. Whenever I saw one on the sidewalk, which is pretty frequently around here, I would just always have vials in my backpack, or grab it, take it to work the next day. And so this next video is an adult female smoky brown roach doing the exact same jumping movements in the same situation. Very graceful. And they did the exact same behavior. So from the start of the jump to the end of the jump, it took a bit longer, but they still did it in the exact same movements. So they were pushing off with their middle legs while they moved their hind legs forward. Then they were pushing off with their hind legs until the middle legs left the ground. And then they were completely pushing off with their hind legs until they left the platform. And for like a visual graph of the series of movements, this is what it was. Sometimes they would pair it with a wing flap, sometimes they wouldn't. If they flapped their wings, it was after they left the platform, so I didn't label that as like a contributing factor, but we'll get to that as well. And here's some of my data from this set of research. The speed at takeoff was not significantly different between the lab colony and the wild-caught roaches, nor was it takeoff angle. So the angle that their body was with the platform when they were off of the platform, it was almost the same. However, we've still couldn't definitively say how much the hind legs were contributing, even though I saw in the series of motions that they move the hind legs forward and then they push up with their hind legs. Maybe it wasn't really doing that much. Maybe it was mainly the middle legs from earlier in the jump. So I obstructed them. This is with American roaches. I would place the roach in a fridge so that they would calm down and let me handle them. And I basically super glued plastic to the bottoms of their feet, which, as you can see here, they slide out from under them when they try to jump, and graceful fall to the ground. The roaches are completely fine afterwards. It doesn't impact how they live, but it does impact how they jump. Whoops. It does impact how they jump. It might be in click order. No, it's not. And for the control, I glued plastic to the upper part of the leg that isn't touching the ground, and they jump just fine. And here's the data to support that. There is a significant difference between the control roaches with the skis on the upper part of their leg, and oh, that's the treatment. The control with the skis on the upper part of their leg and the ones with the skis on the bottoms of their tarsal pads that make contact with the ground. This is the normal data set. So there was a significant difference between the ski roaches and both the control and the normal, but not between the control and the normal. However, the takeoff duration was still the same. It still took them the same time to perform the jump and leave the platform. It's just the power and their propulsion was affected by their hind legs being changed. However, to add another layer of depth, what if they were jumping in a different situation? This is just single roaches jumping off of a single platform. So, and maybe the wings are in a place somehow. They didn't really flap them that much, but what if I gave them a situation where they didn't need to use their wings at all? So I set out to have them jump between two platforms. These platforms are 3.5 centimeters apart. And this is an American roach male. And so they, same situation shown on with a bright light. And oops. No. And they jump in between without needing to use their wings at all for the jump part. They still kind of put it out like an airfoil when they're landing to drag themselves down, but they don't use it to propel at all. This is in a smoky brown female that doesn't even open them as she crosses the platform. And here's the data for that. There were no significant differences for the speed between the normal single platform and the gap jumping platform between either species. There was a significant difference between the takeoff angle for Americans, but not for smoky browns. So from my research, I can make a few conclusions. That American and smoky brown roaches in the genus Paraplaneta, they have a stereotypical behavioral jumping pattern. They use their middle legs, they move their hind legs up, then they push up with their hind legs until they leave the platform. When you change something to their hind legs, their jumping ability and skill is affected very clearly. And when they're crossing gaps, they do the same behavior, and they don't even try to use their wings. They know that they don't need to. The next step is to put skis on their legs and then have them cross jumping the platform. And I didn't add it here, but I'm noticing a very distinct difference between antennal lengths. So when they can feel the other platform with their antenna, they know it's there and they try to jump for it. If their antennae are shorter because of something that happened to them in the wild, or they're just born that way, they don't even try to go for the other platform. But I can't statistically prove and say that that's what happens. so I would need to research further to find out. I'd like to thank the papers that I use to base my research off of, and the Office of Undergraduate Research for giving me travel grants to be able to present this at different conferences.
And just noting that photograph.
This photograph, yes, this is a still from a video that we took for our YouTube. This is not from a data video. So we took this video for YouTube. And so this is like a frame of it. And I submitted this to the graduate school's envisioning research competition. It got on the front page and I was like, but yeah. Yes.
I have a question about the platforms.
Mm-hmm.
It looks like they're like tucking their feet over the edge of the platform when they're launching off. Do you think that you'd see that same like sequence of behaviors if there wasn't that platform shape, like where they're just on a flat surface and wanted to jump off that?
I think I definitely would. This is before they even started jumping. So in all the jumping videos, their front legs are like stretched out. So, but definitely sometimes the middle and hind legs are over the corner a lot. If they were really on the edge, I ruled that video out. So I wasn't using those, but it definitely could play a part if they have a firmer grip or something of what they were jumping off of. Yeah.
Jumping distance at all, or do you think that would have any indication of like how the hind legs play into this jumping behavior?
I haven't looked at jumping distance, no. I know how far they jump because I know how many pixels long they're traveling, and I know how far a pixel is per video. But no, I would suspect that that's very dependent on the sex of the roach and also their size and weight. On average, I've noticed that the female roaches are a lot heavier, especially if they're pregnant. So they not only don't want to jump, but they fall more. So I feel like jumping distance would have a higher correlation to body weight than skill, but we could definitely prove that. Yes.
You know that these actions are not sort of the stereotypical actions that they use start flying, and they just decide to fly.
I don't. I would suspect that this is exactly what they do when they want to fly. I think that very rarely do they fly from level surfaces. They're mainly flying off of like vertical surfaces. So because they're on a level surface, and especially for the gaps, they know that they're going from one place to another. They jump with the intent of jumping, not jump with the intent of flying. I can't say what's in their mind, but I could definitely see if their jumping behavior is different when like inducing a flying behavior instead. I feel like it would be the same, but I'd still have to prove that.
I think would happen if you glued their wings shut.
I don't know. I feel like if I glued their wings shut, many of the jumps would look like this one, where they don't even try to use their wings. I feel like it would look exactly like that. But I don't know. Cool. Thanks.
Up next, we have Cole Hankinson.
So I'm Cole Hankinson. I'm a plant biology student here at NC State, but I also have a minor in applied ecology and in entomology. My project was the floral visitors of native and invasive Symposium, basically within North Carolina. So thistle ecological relationships have been looked at for quite a long time just because they're so pervasive around the environment specifically. They attract a lot of pollinators usually, and they're quite ecologically significant for a large number of species. And obviously speaking, the differences in pollinator dynamics can really show either what incentives insects have for visiting those plants in general, and what incentives those plants in turn want to give out. The only problem with that is, obviously speaking, invasive species can kind of put a whole wrench into that. And since insect pollinators are critical, very critical in terms of how environments work, any wrench in this can disrupt native species that might fundamentally rely on a handful of species. So the objectives here were to, one, find two different Cirsium species. So the native one here is Cirsium horridulum here. And the invasive one here is Cirsium vulgare. They're pretty related with one another, though they cannot breed, which is rather different species. The aims of the project were then to collect a consistent number of floral visitors specifically. And the reason why I use floral visitors here is because just because something visits a flower does not mean it's a pollinator at all. So secondarily, we need to analyze if pollen was found on any of these floral visitors to see if transmission and fertilization is possible then. And if they carry a certain amount of pollen or just pollen at all, then it's at least more likely that these floral visitors are then pollinators. And my general prediction was that Cirsium vulgare will have more floral visitors with pollen than Cirsium horridulum, as according at least to literature and I suppose my anecdotal experience, Cirsium vulgare is very common within North Carolina, and Cirsium horridulum is one of basically the most pervasive native Cirsium species in North Carolina, but it's not found in the same amount of places, at least. Those are the suggestions that I've read. Methods. So I had to choose a shared field site because any geographical changes between them might have a bias in pollinators. So this was a young pine stand located at Shank Forest, and where significant populations of horridulum and vulgare were both present, and insect samples were collected opportunistically from each. The populations basically grew in sort of like stands around each other. So I would go and collect a few insects from a stand, move on, more or less, because obviously collecting only from one stand is going to present significant issues in terms of diversity. And this is just where it is. So it's in this like middle area here. It's a cleaved out section of Shank Forest here. The methods were as follows. So I collected a total of 50 insect floral visitors from each. These insects were then euthanized using a kill jar, identified, pinned, and labeled. In the lab, their bodies were either swabbed with wet paintbrushes, or sometimes they just had so much pollen on them that I could just like take a needle or something like that and swab it on there on a slide, which would then be analyzed using a light microscope to see if pollen was there. And there was pollen. So these are Cirsium horridulum pollen grains at the top and Cirsium vulgare pollen grains at the bottom. There's actually a lot of diversity in pollen form here, although they all have like these little air sacs, which is interesting. So some of it was not as clear as initially implied. Luckily speaking, it appeared to be that in the field, Cirsium horridulum and Cirsium vulgare bloomed at very different times. So Cirsium horridulum bloomed first and then died. Cirsium vulgare then bloomed after that, except for like one case that I saw, more or less. So anyway, within these populations, these insects were then categorized. If pollen it was present from the plant they were collected, if pollen was absent, or if unknown, if pollen was present. This could be like ambiguous or just like pollen fragments, where I can't definitively say if it's that, even though I really do suspect that it is. So the results. This is an Osmia species, which will be important soon. A total of 29 out of 50 insects on Syrsium horridulum, or about 50%, were found with likely horridulum pollen, and a total of 39 out of 50 insects collected on Vulgare, or about 78%, were found with likely Vulgare pollen. So the prediction was correct, more or less. More floral visitors to Vulgare were found with pollen, though I will say in this that out in the field, at least to this one stand, Syrsium vulgare produced a lot more pollen than horridulum, as I took flowers from both and swabbed them to collect pollen. It was very significant, though obviously I have no definitive way of counting the individual pollen grains without making myself go insane. So of the 29 horridulum insects, about 10% were hemipterans, which were mostly came out to leaf-footed bugs. Hemipterans are defined by their piercing, sucking mouthparts. 20% were coleopterans. These are beetles. And about 70% were basically just bees, more or less. And of these, interestingly enough, osmia bees, which are, at least in literature, said to be a specialized pollinator of Syrsium species specifically, made up about a third of all insects with suspected pollen. Now, they were probably Osmia chalabi. I hope I pronounced that right. I can't definitively say for certain, because there's another that's very scattered around here called Osmia texana. It is mostly found near the mountains. There have been scattered instances here, and they're almost morphologically indistinguishable. But I can suggest that they were probably chalabi, but I can't necessarily prove it. But anyway. As, however, for Vulgare, about 13% were hemipterans, 51% were bees, and I believe there might have been an ant in there. Hemipterans constitute ants, bees, and wasps, specifically. And 23% were coleopterans, or beetles. About 2.5 were dipterans. I believe there was one. These are flies. And 10% were lepidopterans, which constitute butterflies and moths specifically. So as you can see from the data, we have very interesting statistics, actually. So it seems to be that the Syrsium vulgare pollen carriers are a lot more spread out than the Syrsium horridulum pollen carriers. But in that, there are still notable pollen carriers. Interestingly enough, Japanese beetles actually carried pollen, even though they're plant feeders. There are a lot of American bumblebees and common eastern bumblebees, a lot more than the horridulum in this. And this could be just lifestyle specialization. American bumblebees and common eastern bumblebees are generalist pollinators. They will basically go for anything that they can recognize that has pollen, which means that, at least in this circumstance, vulgare, this is probably how it's spread so far, right now. But for horridulum, we have two main pollinator populations, Osmia, with about a third. And interestingly enough, there are actually zebra longhorn beetles that carried this pollen. I did find them within inflorescences, and I could not capture this with a camera or anything like that, but I did see them make kind of like sweeping motions on themselves, which would at least suggest collection of pollen in some capacity, at least. And there were American bumblebees, common eastern bumblebees, but they were not as necessarily common. So for the discussion, this is a zebra longhorn beetle, by the way. They're quite small. So for vulgare, again, appear to attract a wide variety of known generalist pollinators, such as common eastern bumblebees, which were 8 out of the total of 39, and American bumblebees, 7 out of 39, and eastern swallowtail butterflies, for instance, indicating a more diverse pollinator community than Horodulum. Horodulum, though, however, has this specialized community of insect floral visitors, specifically with regards to osmia bees, and even suggestive of zebra longhorn beetles in some instances, even though, according to literature, are very disagreed about what zebra longhorn beetles actually do. So this would at least suggest that even though vulgare is spreading around, Horodulum might actually not be that threatened, at least in this specific instance, as they're growing right next to each other. They seem to coexist quite fine. They flowered at different times, and the specialized pollinators likely will not jump over for at least a long time without a long time of a specific evolutionary change, which we cannot much predict, at least. And though in this, I will say, certain notable limitations exist. Again, the possibility of pollen lookalikes on floral visitors is always a possibility. Since these are both Asteraceae plants, it's always the technical possibility, even though that they can't cross over, that other Asteraceae pollen grains that just happen to look a lot like Syracem pollen grains have landed, or that I collected pollen grains from them, because some only had a few pollen grains on an individual insect. The possibility that, the fact that this is just one area, more or less. So obviously there's a sampling bias there for one summer. Some insects were harder to collect than others. I did see a couple of, for instance, butterflies that I could not collect off of Syracem Horodulum, just because their flying pattern was very evasive, let's just say the least. So unfortunately, even though I saw them about 2 or 3 times, I could not collect them. Others, for instance, were on broken ground, and others I just saw once and I just couldn't collect them, more or less. And the potential differences in pollen production, obviously, between vulgare and Horodulum. For instance, perhaps in terms of attraction, there is really no big difference, but just because vulgare appears to, at least in my experience, produce more pollen, it just attracts more generalist pollinators, even though theoretically speaking, if this was a stand with only Horodulum, maybe the American bumblebees or the eastern bumblebees would visit it just fine, more or less. And obviously speaking, a study across a wider environment is necessary to draw super definitive conclusions, even though this is more or less a good pilot study. And additionally, a machine capable of categorizing and differentiating pollen would expedite this research significantly. It turns out that it is very hard to look at pollen grains by hand and tell exactly what they are. Yeah. But I would like to acknowledge Dr. Sorensen, of course, for all his help and recommendations through the course of this project. It would not exist without him. Are there any questions at this time? Yes.
Very cool stuff. When you find that machine, let me know.
Yeah.
And you're not the only one wishing that. I saw that you had the 2 bumblebee species visiting both types,
Mhm.
but only osmia on the one.
Yes.
So do you think that that's, and you were saying that they did not, or very little overlapping of bloom times.
Mhm.
Do you think that osmia might be driving the speciation of their phenology? Or is it that just that osmia is emerging later in the season,
Mhm.
and so are just found on the one species and not the earlier period?
Well, in this, Horridolum actually blooms first, interestingly enough, despite their larger inflorescences, which tends to be the opposite in some cases of plants. But it's actually the fact that Vulgare is actually a very recent addition to North America, more or less. I believe they're mostly native to the European, North Africa, Asia. Vulgare is not even native in this instance. So yeah.
I guess related to that, for those of us who are not plant people, like
Mhm.
we saw the pictures, but like how different like are the flowers like
Mhm.
from an osmia perspective? Like can
Yeah.
an osmia
Mhm.
is it that flower or is it like super different than what they're used to?
So theoretically speaking, in terms of a physical disposition, they would be able to visit. I heavily suspect in this that there's some other factor, whether it's olfactory or some other chemical cue, that makes osmia want to visit Horridolum. So for instance, Horridolum, it's a lot more cone-shaped with the inflorescences are inside that cone. Their inflorescences are bigger. The plant tends to be bigger. Hang on, I can go back all the way. Oh. The size of this plant is not done justice by this photo, let's just say. And Vulgare, it's still significant in terms of its size, but it is not quite as robust as Horridolum. And as you can see, Horridolum sort of has this central stem from which flowers radiate kind of like outwards, while Vulgare, it more splits out in this. But to answer your question, there's technically and as you can see, Vulgare, their inflorescences are a lot more just out in the open. But technically, there's no physical limitation to answer your question. It is probably another environmental or biological factor.
What like methods did you use for collecting your insects?
Random and opportunistic sampling, more or less. So I used a sweep net in order to collect them, as I did not want to be stung. But in this, it was random sampling between plots with switching in between plots, more or less. As initially, I had thought about doing a timed factor where I would just collect everything at a certain time, but certain insects are active during certain times that creates a bias as, I cannot go out in the field at certain times. So that would create then a bias. But of course, it's very hard to conduct studies like this without at least some bias, of course. Yes.
Sweetness.
Mhm.
You're getting certain connection.
So in this, I was actually very careful with collections. So I would only collect insect species that I saw went into the inflorescence. And I would wait for them either with a sweep net. A lot of the times I would actually just use a jar and that was simple enough. But yeah, I was very careful about cross-contamination because there were also a significant population of leaf-footed bugs there. And I just did not collect any leaf-footed bugs unless I saw them right on top of the inflorescence, which they did sometimes. So,
Finishing out this first session, we have Jordan Holcomb.
Hi, I'm Jordan Holcomb. I am studying plant biology, but I grew up at the beach and have an interest in fish too. So this is about my experience at the North Carolina Aquarium on Roanoke Island, which is where I grew up. So that's how I ended up there. And I studied blue gobies and how water quality impacts their aquaculture rearing success.
So basically every aquarium is assigned a species in North Carolina, and the one on Roanoke Island is the blue goby. And programs can distribute their species to each other, or they can trade, or they can sell them to other aquariums across the country. And it just creates a more sustainable system so that we're not catching them from the wild, because they can bring in parasites and disease and impact the ecosystem that we're removing them from. And this is the father of all of the baby fish that my experiment was on. And you can't really see it, but there are eggs up in the top of this tube. They lay their eggs in the tube, so we put PVC in there so that we could remove them easily and take the eggs away, because if we did not, the father would eat all of the babies right after they hatched. So he was very protective of his eggs. So blue gobies are considered an easy fish, but the aquarium was still struggling to raise them to adulthood. When I joined the team doing aquaculture, they had been trying to get them for about a year, and they still had not gotten, they'd only gotten one single fish to adulthood in the whole year that they were trying to do this. And they brought me in and they were like, fresh eyes, fresh mind. And then we wanted to try a bunch of new things to see if we could make the babies turn into adults. In the past, they were able to get the fish fry, which are the little itty bitty babies, but they couldn't get them to morph into adults. And you can tell that they morph when they have a blue stripe on them, because before they're kind of clear and then they get a blue stripe. It happens overnight. Very cool. Then when I joined, they were getting eggs, but then they would disappear before they would hatch. And to test that, we started removing them earlier, but then there was less success of them morphing into adults, all kinds of issues. And then after my experiment, we were getting eggs, fry, and adults. So this is their food source. They eat rotifers. So we had to grow the rotifer colonies to feed them, and then we had to count rotifers under the microscope, which was really annoying, especially when you have horrible vision like me. And then the baby fish don't have very good vision because they have just hatched and their eyes haven't fully developed yet. So we had an algal colony also that we put into the water so that there was kind of like a difference in color so they could actually see the rotifers to eat them. So we hypothesized that the water quality was what was killing the fish for two reasons. One, the ammonia levels were too high, and ammonia is toxic to fish. We thought maybe it was coming from their poop, and that we were putting too much food in there. So to combat that, we did more frequent water changes to remove the ammonia water out and put new water in that was clean. And the second reason was the salt. We didn't think it was a high enough quality for them because they're so sensitive. So we switched to a different brand that was more pure. It had less chemicals in it, more natural type stuff. And we also introduced that through the water changes, because you take the bad ammonia water out, you put the new water with the more pure salt in. And you can see this is our increase in water changes. We used to do 1 to 2 times daily, 20% water changes. And we increased them to 2 to 3 times daily, 20%. We tried to be as consistent as possible, but there were multiple people working on these systems, and there's a lot of things going on at the aquarium. You can't always get to it 3 times a day because that is a lot of work. There is definitely a positive trend. And we switched the brand to the same one that they use for the invertebrates and the jellyfish, because they are also very sensitive and had more success when they were raising their larval jellyfish. So these are the babies. They're so tiny. And every single day I had to count all of the babies and all of the nests. It's not possible to count all of the eggs because they're so small and they're in the tube, and we didn't want to disturb them by taking them out and putting them under a microscope. You can kind of see, like, this one has morphed. It has a blue stripe, this one, but then these down here, these ones are younger, and so they aren't blue yet, so they're still fry. We counted them daily, the fish, the nests, and the adults, and kept track of those, and we kept track of how often we changed their water. And it worked. So this shows how our fry are surviving longer. When I first started, they had one singular fish. This whole green line, that's the one fish that they got to adulthood. It was kind of sad. It was like this whole big tub and one tiny fish in it for months. Poor guy was very lonely. But then all of the other nests, they would hatch, and it was a very low success of hatch. You can see that they only had like 5 fish that actually hatched, and then they would die a couple days later. And then by the end, we were getting almost 100 hatched eggs, and then it's normal for them to kind of come down, but then they stabilize after a few days. Especially when they start to morph, it's about the time that their swim bladder starts to form, and they're more likely to die around that time, just natural causes. And we also had so many eggs that were hatching that we had to create tub 4. When I started, there were only 3 tubs. We had to create a new one because there were so many fish that were living. We also had more eggs that were hatching. You can see there's a positive trend right here in the eggs that are hatching. Because when I first started, the eggs were just disappearing or just not hatching at all. And they just started to hatch more. You can see our survival rate is 30%. When I started, it was about 1%. So that's a good increase. 30% is still kind of low, and we're trying to work on that too. And this lines up with this chart if you want to see when they're dying. And like I said before, we had more active tubs. Before they would only live a couple days, and then they would die. This is again that singular fish, poor guy. And this blue line also was a singular fish. So then here's when we started to get more numbers. And we also had more frequent nests. So this shows the positive slope of our numbers of nests, and all of the blue dots are ones that survived, and all of the ones that are red are ones that died. And even though we still had some nests dying towards the end, they did live around 50 days, which is about the time that they morph. Like I said before, that dying around that time is normal, but before they were dying at only 1 or 2 days. So this is the breeding rack. These are our 4 breeding pairs. And we learned that aquaculture requires meticulous attention on water quality standards, which should be implemented across the board. And regular maintenance is not sufficient. It requires a lot of training to be able to keep these fish alive because they're such sensitive creatures when they're so small. And then one issue we had with the study, all of the eggs were from this breeding pair. These 3 did not lay any eggs the entire period, and we don't really know why. We hypothesized that it's because they're old, because they had these breeding pairs for a couple of years when they've been trying to do this in the past and had no success. And we wanted to look into getting new breeding pairs to replicate this and see if they've just gotten too old to breed. And it's also important because we don't want to have inbreeding if we only have one breeding pair making all of the fish. I was talking about ammonia earlier. This is the test we use for ammonia. The green means it's bad, it has ammonia in it, and the yellow means it has 0 ammonia, which is what we want. So we would test that regularly and make sure that it was at 0 because of our water changes. When I first started, it was green, so that's probably why they weren't working the whole first year. We want to manipulate the water quality to test other breeding pairs, since, like I said, all of the results were from one breeding pair. And we also have some new fish that they had just got in when I left, they had both captive bred and wild caught. So we want to compare if there's a difference between those. And I don't have a picture of my mentor, but he did give me this cool lamp. So shout out to George. He was really good. It has feet. I don't know if you can see, they're really funny. It just showed up on my desk the last day of work. Any questions? And that's me releasing a sea turtle.
Here's a question.
Okay.
Do you think that there might be other factors or limiting resources that are kind of limiting the production and survival at different life stages? like, could it be density dependent? Do you need like even more tubs so there are fewer fry per tub?
I don't think so, because it was just kind of consistent that around 50 days is when they would just start dying, because that's when they start morphing and developing their swim bladders. And they also have different stages of their life when they eat different things.
Mhm.
So when they first hatch, they eat nauplii, and then they switch to the rotifers. But when they switch food sources, sometimes they just don't, and then they starve to death. But it's really hard. You can't force a itty bitty microscopic fish to eat. So whenever we had food source changes, they would also die then. Yeah, I think it's just natural. I don't think it's a limiting.
So you're
Any,
just facing typical like our strategy
Yeah.
challenges.
Are you gonna continue this work?
so I was an intern at the aquarium between my freshman and sophomore year, and then between my sophomore and junior year, they just texted me in January and they're like, "Hey, we have an open position. You want to come back?" So then I went back. So I don't know if they have another job this summer. I grew up on the Outer Banks, and so I just lived at home and worked there. So I'm not sure what I'm doing this summer. Okay. I'm actually working at the Yates Mill lab right now, and we're doing aquaculture of snails and mussels. So I am still doing aquaculture. I just don't know if I'll return here. And now that they're having more success, I don't know. They're getting plenty of fish now. Like they have too many, and I don't know if I said this tub. Oh, wrong slide. Oh, there's so many. This tub is where the babies go, and right now it's full. There's just so many, and there are this one breeding pair. They're just busy, I guess. I don't know.
You worked yourself out of a job.
Yeah, I did. Mhm. Okay. Okay, thank you. Well, they live a couple years. Not like a long time because they are fish, but they do live a few years. None of them died while I was there, so
Thanks, Jordan.
thank you.
This is part 2 of 3 for the fall 2024 Applied Ecology Minors Research Symposium. Without further ado, we'll move on to focus on, highlight, and celebrate the achievements of our student presenters today. First up, we have Sally Thompson.
Yeah, so I had the great opportunity to work this summer as an REU researcher in Puerto Rico, specifically focusing on flood disturbance on the assemblage composition of macroinvertebrates. So just to dive into some background, precipitation happens in all different levels throughout the world depending on the biome that you're in. And natural disturbances like hurricanes, droughts, or floods could cause streams or lakes to periodically dry up or flood. And as we all probably know, climate change is making these events happen more frequently and more intensely. Specifically in tropical rainforests, they receive an average of 2.5 meters of rainfall per year. And they're also characterized by varying gradients and by shallow root systems, which prevents water holding capacity in the soil. To jump ahead real quick, this top photo is a topographical map of Lucia experimental forest where I worked. And you can see that these mountain ranges make like a perfect example of a watershed just funneling all the water down into the streams below. And so to hone in on the research and the importance of it, macroinvertebrates are very important to stream ecosystems. These organisms aid in nutrient cycling, as you can see in the top photo by the very frilly lungs on this macroinvertebrate. They also aid in population control, as seen in the carnivorous dragonfly larva below. And of course, so if these macroinvertebrates are in danger, if they're affected, then these ecological factors will go kind of haywire. And so, as alluded to before, my study site was in El Yunque National Forest on the eastern side of Puerto Rico. Its elevation ranges from 350 to 500 meters. It has an average rainfall of 4 meters per year, which is higher than the general tropical rainforest average. And then we specifically worked in the Prieta stream, which is made up of these two tributaries. Prieta A was our control group because it just has a natural flow. And then Prieta B has a 30% reduced flow due to a system of PVC pipes that were put in a few years ago. It starts at the top of the arm and then just diverts some water down about 130 meters. And so this part is going to sound a little jumbled. I hope it's clear, but I specifically used, the streams are quantified by individual pools. And I specifically used 6 pools in all, 3 per treatment, to place my mesh pouches, which were used as the artificial substrate. Hopefully that made sense. And so this is what those pouches look like on the left once they're placed in the stream. And so my experiment was conducted over 5 weeks, collecting 1 pouch per pool each week. These pouches averaged a volume of 148 milliliters and a surface area of 346 centimeters squared, just for reference. These right 2 photos just are some from the field, just to show the collection process and being sure none of the critters were like stuck in the net when I collected these. And then all of these pouches were put into bags and labeled accordingly. Once at the lab, the bags were emptied, the substrate was rinsed thoroughly, and basically anything that moved was collected and put into vials of alcohol to then later be observed under the stereoscope to be identified to family level, generally counted, and then also assigned a functional feeding group. Some examples here are fly larva, caddisfly larva, and then also a mayfly larva, which is the most abundant thing that I ended up finding. And so once all that was collected and recorded, the data was compiled in a couple different ways. First, it's shown in line graphs, and these were measured by first just chronological collection dates, and then also in days since the last storm. So, for example, this collection date, there happened to be a storm or disturbance 2 days before that collection date. And then for reference, Prieta A is shown in orange and Prieta B is shown in blue. And these were all measured in abundance, biomass, and richness. They were also broken down a bit further into functional feeding group to try and see a correlation in that. And this is represented in bar graphs. For space sake, I've only shown Prieta B. I have 12 bar graphs in total though. And so out of all these data sets, only 2 showed a significant correlation when ran through linear regression analysis. Our R squared value was set at 0.5 or greater, and our P value was set at 0.05 or less. So anything meeting that requirement was deemed significant. So the only thing that was significant was that Prieta B showed an increase in richness over time, and then collector gatherers showed a decrease in biomass over time. And these were the graphs on the linear regression software. So my original hypothesis was that the abundance of insects will decrease soon after flood disturbance and water levels, and different functional and taxonomic groups will respond differently to flood disturbance. due to the lack of significance, I did have to reject both of these hypotheses, which, as we probably all know, just happens in science. It's trial and error. But that doesn't mean that the experiment was fruitless by any means. So some contributing factors or some future explorations we could think about when trying to conduct later research or just general reflection are the fact that some of the rock pouches, the substrates got buried over time. And so just maybe honing in on trying to perfect that procedure and that setup would help. Also, we ended up qualifying what deemed a disturbance after we got all the data together, which helped us. But it might be better to maybe qualify that beforehand and maybe using this experiment as a way to do that. And then the last thing that really stuck with me, and I think definitely impacted our results, but we didn't end up using as like a variable in our data, was the amount of storms that happened before each collection date. So this is the graph of water levels, and each peak is like a disturbance. And so as you can see on the right, a lot more storms happened before the collection date versus these other collection dates, which are shown by the green arrows. So yeah, that's the gist of my experiment. I really enjoyed it. And I just want to thank the National Science Foundation and the RU Network for funding my projects. And I definitely could not have done it without my mentor, Dr. Ramirez, and my RU coordinator, Momery. They're in the back. And then also the technicians that were down there and the rest of my research team. So yeah, thank you for your time, and just let me know if you have any questions.
To like design a follow-up experiment, you just told us like there's some confounding variables. What would you like most like to explore and maybe control for?
Probably, like I said, more focusing in on using the amount of storms that happened as like an X variable, because I think that definitely impacts the recovery ability of the populations and things like that. Yeah. Yes.
I was curious, and I think this builds off of that question, and you alluded to this a little bit during the end of your talk. I'm thinking about the pouches that you use. Are there different types of pouch shapes or the way that they're placed that you would test out to maybe see if you capture another sector of the macroinvertebrate community, or do you feel like you really did capture much of that community in your sampling?
I hope that we collected enough. We kind of made them all the same shape. Let me go back to a picture of them. Yeah. Oh, nope. Nope. There we go. Okay. So I made them kind of all the same shape and tried to make them uniform just for consistency purposes. I did kind of like zip tie them all together so that way when disturbances did happen, they'd be less likely to be washed downstream. Some of them did end up getting buried like the last week, because again, there was a lot more storms. But I would hope that the substrates did do a good job at like allowing a lot of different like feeding groups and different richness to inhabit that. There's also like freshwater shrimp in the streams that the other research team dealt with. And so hopefully the mesh wasn't big enough for them to get through, but it's a possibility. I did end up getting a couple in my nets, so. Yes.
It may be that the data, the answer
maybe the equality of the rocks, you know, those
Mhm.
were new rocks that we purchased in Home Depot.
Yeah.
But they didn't have anything growing on them. They were clean rocks.
Mhm.
So maybe some pre-incubation, so they grow some algae and some biothings like health indicators. So they may be more effective for insect colonization.
Yeah, that definitely helped. Yeah. Like you said, we just kind of bought them at Home Depot and then I just like scrubbed any dust off of it. So they're pretty clean. Not a lot of stuff already built up on them unless it was like week 5, but yes.
Going back to, how do you know if you fail to capture certain species?
Mhm.
Right? Is there a database or literature that you can rely on to say, this is our expectation of what we should find at this place at this time, and then compare that to what you actually find, so that you know if you're missing something?
I'm sure there is out there somewhere. I did end up using a database from Dr. Ramirez that was basically a table of all the species that they have ever found in that stream. But it wasn't, I believe, like quantifying or the ratio of them. So,
Right, at least that.
yes, yeah. And that's what kind of helped me identify which functional feeding they belong to as well. So like I said, there was definitely a dominating one.
All right, thank you.
Thank you.
Up next, we have Isabelle Quadt.
All right. Thank you all for being here today and coming to listen to my talk. So my name is Isabelle Quadt, and this summer I had the opportunity to work here at NC State in the biotechnology program with Dr. Hayden Hudgens and another undergrad student, Patrick Smith. And we studied the differential localization of a germline specific E1F4E paralog to stress granules in response to heat, osmotic, and meiotic stress in C. elegans. So a little background on C. elegans is they are about 1 millimeter long and they are about 88 micromillimeters wide. So they are microscopic, and so you have to use a microscope to look at them. So as you can see, they have 2 little arms. They're called gonad arms. This is where their germline is, and that is what we focused on. So here is one of them up close. These worms are hermaphroditic, so they first go through spermagenesis and they store their sperm in their spermatheca, and then they go through oogenesis where they create oocytes and they build up in their arm. And this happens on both sides, so it's not just like one creates sperm, one creates oocytes. It happens in both arms. And so up here in the smaller section, it's called the rachis. And so you'll see that we looked at both the rachis and the oocytes. So these are early germ cells and late germ cells. And then we specifically focused on the translation portion from RNA to a protein level. So why is this important to ecology? So it is a model organism. So its genome has been mapped as long with its life cycle. It has a pretty short life cycle. It takes only a couple days for it to become a full adult and lay its eggs. And then they'll live for about a week or so, depending on what temperature you keep them in. Not much is currently known on what happens to animals when they are put under a lot of stress. So which is why we studied this. So if these granules are forming under heat stress, osmotic stress, and meiotic stress for these worms, it's probably happening to a lot of other animals here on Earth. So it can give us a good idea of what's happening on a genomic level to animals with climate change. So here's my abstract if you wanted to read it, but I'll just go over the cliff notes. So what is known, so stress granules, also known as P-bodies, have been known to respond to the stress of aging, which is meiotic arrest. In oocytes, which are germ cells, and environmental stressors such as osmotic stress, arsenic, and heat stress, proven by several studies, which I reference in my abstract at the end of this presentation. And so the gap in knowledge, though, is we were looking for co-localization between IFE, which is a protein recruiter, and an RNA protein, which is called GTBP1, which is a stressor. And we're looking at this because IFE3 is known to repress translation of developmentally important RNAs. And so we predict that IFE3 would co-localize with GTBP1 for other studies we looked at. So our methods, We created genetic crosses, which I'll go into here in a second of what we did. But we were specifically looking for ones that should have been the germline. So we made several ones that were fluorescent. So all these are already in the worm. We just create crosses that fluoresce certain proteins and the receptor. And so ones that were important were TOM20 and GTBP1. My partner focused on TOM20. I focused on GTBP1, which is why I'm presenting it today. And so for heat stress, we use an incubator first to do the control in 20 degrees Celsius, and then to do the test in 30 degrees Celsius. And they were kept on agar plates, which is here on the left is what this is what it would look like if you're looking at them under the microscope, and you can see how big they are at their different stages. We also kept them in the incubator at 20 degrees Celsius for our meiotic stress, and then we put them in Eppendorf tubes for osmotic stress in different salinity levels. And we also did imaging with an imaging microscope that had fluorescence under it. So to create our crosses, my mentor had already had hermaphrodites that were tagged with green fluorescent proteins on the IFE3 recruiter, and then red fluorescent protein on the GTBP1 protein. And so what we did was we took, while these worms are hermaphroditic, they do naturally have males in their populations. It's just at a significantly less ratio. So we did find a couple males that didn't have the fluorescence. We crossed them with a hermaphrodite to create males that did have the green fluorescence, and then crossed those with hermaphrodites that had the red fluorescent to get hermaphrodites that had both the green and red. We then found the strongest ones that had both green and red, put them on plates by themselves and waited for them to self-fertilize and see which ones had pure, fully green and red offspring. And that was our positive one for green and fluorescence. And so this is what they look like under the microscope under normal conditions, with our GTBP1 being red, our IV3 being green, and then you can see a merged image so you can see where localization happens. So our different methods is our heat stress. So we had about 150 transgenic worms. And we put them on NGM plates, which is just their normal feeding plates with the substance they need to survive, at 34 degrees Celsius for 30 minutes, 1 hour, and 3 hours. And then what we did was, after that, to image them, we took agar gel plates and we put a paralyzer on them, put the worms on there, wait about 10 minutes for them to stop moving so we didn't get fluorine in our images, and then we put them under the microscope. We did the same imaging for our osmotic stress after we put them in H2O, M9, and 1 molar NaCl for 1 hour. We did the same imaging, and same with meiotic arrest, except for these worms had already laid all their eggs, and it was about 3 days after, so they were under that meiotic stress because they still had oocytes but no sperm to pair them with. So our results, here we first have our control for the heat, which is at our 20 degrees Celsius, and as you can see, there is the green is pretty diffuse. Same with the red; it's not, you can't really see it very well. And then when you merge, the green is dominant. And then after an hour and a half is when you first start seeing foci form for both of them, both the IV3 and the GTBP1. However, IV3 is more prominent and kind of dominates in our merge. And you can see that some areas overlap, which is why they are yellow, or but you could adjust the how strong it was, so that way you can still see the red come through even if the green was dominating. And as one thing we found was in the green, if you look up here, like it's more globular and it takes the whole area. Meanwhile, the red had more distinct foci in the area. So that way, so you can tell that they were different and they were forming in different localization ways. And then you saw the same thing at about 3 hours, but a little bit stronger for both. And then in one of our papers, we saw that that IFE3 or GTBP1 would diffuse after being put back into normal heating conditions at 20 degrees. So we did that for about 3 hours to see if it still would diffuse, and it did in the red, but not fully yet. But the paper said it did take several hours to do it, but we just observed it since we already had them made. And then for osmotic stress, we had an isotomic condition, which was our control. So as you can see, both the green and the red are diffuse, with some foci forming in the IFE3. But they get even more globular after in the hyperosmotic stress for 1 hour. And then the red didn't really seem to form foci in our osmotic stress, but it could be due to we need a bit more longer time to be exposed to it. So then lastly, we had our meiotic stress. So these are 1 day old adults. So they are just starting to lay their eggs or haven't laid them yet. So they are fully formed. As you can see, there is basically no GTBP1 beforehand, but there is is diffuse IV3. And then as after 3 days, they laid all their eggs, they had no sperm left, and they had foci forming in their rachis and in the older oocytes, but not as much with the GTBP1. So that's what I just said, but written out if you'd like to look at it. But what does this mean? So like some other things we did this summer is to do meiotic stress is another way to do it is introduce RNAi with a FOG1 strain, which creates female worms. And so they wouldn't have any sperm at all in their lifetime. And so that didn't end up working. Neither did our other RNAi project looking at genes that solely affect the RNA poly A tail. And we ran out of time this summer to kind of like play with it to see what was going wrong after a couple of tries. So what does this mean for the future? So what you could do is test potential co-localization of IV3 related foci in ribosomes, because that's where a lot of like information is coming from. Because as it showed, we did a knockdown with IV3 for our heat stress, and GTBT1 still forms. So that means IV3 probably isn't recruiting it. So that's a little bit interesting to show. So maybe we look at something else that IV3 could be recruiting. And then also we saw different levels of foci formation in the rachis and in the oocytes. So are these germ cells distinctly different? And like, why do we see different localization in different stages of the oocytes? And then also something I want to note with the meiotic stress is that's something they naturally undergo. Like they're not, like that just happens in their lifetime and their lifespan. So that we might not have seen stress granules forming because they're used to that. So they've adapted to that. So they're not as stressed out as say they would under heat or osmotic stress. And same thing with osmotic, it's a little more common in their soils because of like rainfall and stuff like that. So it fluctuates because they live in the dirt. But heat stress, they generally live in a stable climate about 20 degrees. So that's why they might have been a bit more stressed out. But it's important because of climate change to look at that stress. Here's Dr. Hudgens and I at the Science Symposium in the summer where I presented my research. And then these are my references. Are there any questions? Yes. It's not particularly what I want to study in terms of microscopic work. I do want to do genomic work. So like next semester, I'm working with a DNA set to look at like 2 different species, but it was really interesting and it gave me really good lab experience. I really love the lab that I work in, and biotechnology is really important. So like I would be open to it, but it's not my like ideal avenue.
Why would it scare you all too?
No. Yes.
Are there other abiotic stressors that have some addition to those?
Oh, yeah.
What would be your
So one of the ones we didn't really get to do was the arsenic stress, which has been really interesting, or like different acidities would be interesting to look at because of like rising acidity levels in like soil and like water. So it can be applied to like fish and stuff like that. I think that would be interesting to look at because like it's a natural thing occurring. So
Up next, we have Jake Thompson.
I just want to start by saying thank you all for taking time out of your busy schedule for coming and being here today. So this past summer, I spent in Highlands, North Carolina, working as a botanical gardens intern. And during that time, I also did research on the Sarracenia populations at the station. So what I looked at was Sarracenia purpurea var montana and Sarracenia jonesii, which is a federally listed rare species of carnivorous plant, and the hybrid that is being produced of the two. So background on these guys, carnivorous plants, they evolved over time in habitats that are nutrient poor. So they developed to get their nitrogen elsewhere via mostly insects. Some of the larger carnivorous plants can eat birds and things like that. But we won't be looking at those. These guys had a really cool evolution of their flower too that I know Dr. Tarpey can appreciate. They send their flower way up tall, away from the pitchers so that they don't catch their pollinators. And I believe it's only about 16% or something of their pollinators become prey. Let's see. So my hypothesis for this research was Sarracenia plants will exhibit greater flowering frequency and abundance in managed areas compared to non-managed areas due to the potential benefits of favorable growing conditions, reduced competition, and selective pressures. So shown here is myself with just outside of Site 9, which is an unmanaged site, with the tools I used to take my samples, 1 by 1 meter square, a 200-foot measuring tape, and of course myself to do the GIS mapping. Some of my populations were true counted versus some were random sampled. That's because some of the unmanaged sites were so dense and hard to maneuver that we could not get an accurate true count of the plant populations there. So, well then, that's not what I want. There we go. Yes, sorry. Why is this not working? There we go. So for this research, I had to determine the difference between a managed site and an unmanaged site. So what I used for managed sites were the absence of woody vegetation versus unmanaged would be the presence of woody vegetation. As you can see, Site 1 here has a surplus of species like carex. There is a rare species of grass in here, Calamagrostis canadensis. It's a very open site. Even though you can't see the pitcher plants, they're still in there and they still bloom quite well. And this is Site 10. This is a very dense site. As you can see, a lot of the woody vegetation here. Things like this site had a surplus of Hypericum densiflorum, which made a really thick covering over the plants. I would also like to note that all of my sites had very dense populations of sphagnum moss as well. So I used ArcGIS to map my locations. There are in total 9 sites, even though it's labeled 10. There was a 6th site beside 5, but after further considerations, I just created 1 large site. So you have your 3 green sites. Sites 1, 2, and 4 are your managed sites versus your orange sites are your unmanaged. And Sarracenia jonesii, our federally listed species, was clustered, I think this is noteworthy, here along the water, away from any woody vegetation or other things. And as well here in Site 1, there was no, and there was 1 here in Site 10. There were no Sarracenia jonesii anywhere else in any of the sites during the samples or true counts. I think that leads into that our federally listed species may be more sensitive to overcrowding and lack of sunlight. So here are both my true count sites and random sample sites calculated for their population numbers. This totaled 9,371 plants. I believe this number could possibly be accurate. I believe that we need to do more assessments of that to be sure. And I've come to that conclusion because you can see some of our true count sites like Site 4 and 5 or 36 and 43 versus Site 1, which was just a about 100 square foot difference, square meter difference. And you've got 618. So I'm not too steady with those numbers personally. These are the flowers. I did true count every flower there. We got a total of 1,098 flowers that I personally touched and counted. So that was a lot of flowers this past bloom season there. And so using the numbers on the previous 2 slides, I calculated the percentage of flowering plants in each site. And as you can see, sites 1, 2, and 4, which are our managed sites in blue, are producing flowers above 50%. Versus your unmanaged sites are normally below 50%. I would like to note here that site 5 has an abnormally large amount of flowers. I believe this, while there was the presence of woody vegetation, most of it was Rosa palustris, which is your swamp rose, which has a pretty open growth form, which I believe allowed light to penetrate down to the ground level better. Let's see, versus your sites like 9 and 10 had really dense vegetation such as Hypericum densiflorum, as previously mentioned, and Spirea glabra and Spirea tomentosa and other densely formed species. Here you can see that sites 7 and 8 had 0 flowers. These plants also presented with 0 ethnocyanins. The only plant in these sites were Sarracenia purpurea var. Montana. And purpurea is known to have the red stripes and veins, and these had 0 and 0 flowers. Both of those sites were pretty hidden. I stumbled across them while I was scouring the river or the lakeside to find these sites. And they're densely covered with Rhododendron maximum and Calmia latifolia, which keeps it in deep shade all year long. So with these numbers, I believe that Sarracenia needs an abundance of light. Maybe not necessarily the absence of woody vegetation, but it does need an increased amount of light, like to flower. And here we have the flower. Our challenges that I encountered were the dense areas that made it hard to get a true count. I think that would have given us a more concrete number for this project of plants. I think we can use this information in the future towards conservations of these montane species. So my proposed management strategies for this is going to be the removal or thinning of the dense vegetation if some of these sites is what the biological station would like to preserve. I think we need to remove the hybrids if we're worrying about conserving a federally listed species like Jonesii. I think that a control burn could possibly benefit both flowering number and make it easier to count. But given that all of these are in a boggy wetlands, that would probably be a pretty hard feat to do. And as you can see, I've included this picture. This here is your Rosa palustris, and you can see its open form over a Sarracenia purpurea. So in acknowledgements, I'd like to thank Dr. Stephanie Jeffries for being my research mentor. And I'd like to thank Dr. Erin McKinney for organizing all of this and making it so easy. And I would like to thank Highlands Biological Station, specifically Jim Costa, Jason Love, Mike McMahon, Tara Henderson, Tara Henderson helped me with the ArcGIS. I had no idea how to use that. They were a godsend to me on the ArcGIS. I'd like to thank the Garden Committee and volunteers for all their information that they've done over the years. And lastly, I'd like to thank my friend Alex Waite, who spent Saturdays taking my pictures. So, any questions? Yes.
So what do you think it would take to validate your random sampling with the true counts? Like, and would it even be worth the effort? Because you said you didn't quite press the numbers 100%, right? So yeah, do you think it'd be worth going out and like for one of those patches, getting a true count? Like, do you think it's
Yeah, I
possible?
think if we had the time and, enough people to help, you could section it off and get a pretty good count. I just think that it was very dense, especially site 9 was incredibly dense. You were knocking down things to try to get through it. It was also the largest site that I sampled. Yes.
I just said this and I missed it, but how do you get higher than 100%?
That was funny to me as well. Yes, so what I did is I took the numbers of the population here and divided them by the number of flowers in each section. Yeah, by the plants. Well, I'm sorry, flowers divided by plants to get these numbers.
So that's interesting, because
Yeah.
that means that some plants can shoot up. Like, what's the maximum number of flowers in a single plant?
That's a good question. This right here is probably a cluster of 3 plants, and we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 or more flowers on those 3 to 5 plants that are right there.
Big local distribution, right? Some of them just.
Yeah, some of them really flower and some of them don't. Yeah. Yeah.
That's very kind of autonomous.
Do you think that reflects differences in like the flowers' genetics or differences in soil quality or differences in like those?
How many bugs they caught.
Right, exactly. There was a swarm there and now it's got enough nitrogen to actually promote flowers.
Yeah, I wouldn't think that it would reflect the genetics. This picture was actually taken, Tara and I actually went through and kept this section. This is the display bog out on the trails section really open from the time we got there. And I think it really helped the number of flowers that were coming up. And I think it has a lot to do with sunlight over anything else. Yes.
How did you define a plant? Clone or?
They were clumps, yeah. Clumps. Yeah.
I think this is something I don't remember, but does the station, are these hybrids naturally occurring or like are all the plants at the station planted? Like maybe you go back to your map. Have they spread or are those all like little planted? Are those all planted sites?
From what I understand, there is no record of Sarracenia purpurea var montana coming to Highlands Biological Station. So they believe that it predates the station. I have emails from the archivist and Highlands that sent me letters from one of the directors and one of his friends that sent him several species of Sarracenia rubra, many of them that have been recategorized under different names. Like there was a Sarracenia warei, which we know is not warei anymore.
And it's the Jonesii, the endangered ones that's planted.
That one was not on the list either, but given the date that they sent us, or that they sent the station those flowers, I believe that Jonesii was probably renamed later. I would need to look at that to be completely sure, but I know a lot of the ones on the list were renamed when I got to looking at them. Yes.
So one of your recommendations was removing hybrids or color hybrids from the site. Is that because they're suspected to have some sort of competitive advantage against Jones?
I don't know that they'll have a competitive advantage. There is, down in the coast, there is a hybrid between Sarracenia flava and Sarracenia purpurea. That's Catesbii, and I don't know that it has any significant effects on the ecosystem there, but I think the goal of this project was to see, the population numbers of flowers and try to conserve the federally listed species and, your natural ones. So if you're going to conserve a species, you would want to get rid of the hybrids. There is research being done on the hybrids at UNC Asheville. There was a girl that did her master's on it, and there's a poster available if you would want to look it up. You could get in touch with UNC Asheville.
Dave, do you think there's anything too? It sounds like you found the Jonesii kind of in the wet margins.
Yeah.
Is it really just sunlight, do you think?
It could be a hydrology factor. A lot of those sites were really wet, especially site 1. There was actually a dense patch of carex here that looks like land, but it was submerged. So there's a big hole here that didn't have any Sarracenia at all. But yeah, I think sunlight plays a key role in it. Given the site, the Jonesii that was found in Site 9 that was farther away, it was being overgrown, and I may have forgot to mention that. It was overgrown by Hypericum, and it was very spindly, and you really couldn't almost tell what it was. It had no flowers or anything, so I think they are quite sensitive to sun.
Thank you so much.
Thank you.
Up next, we have Katie Savant.
Hi everyone. As she mentioned, my name is Katie Savant. And so I was looking at larval fish entrainment at the Brunswick nuclear plant with my water resources internship at Duke Energy. And so kind of to start us off with some background and information about Brunswick, the nuclear plant is about 40 minutes south of Wilmington, and it's a relatively unique nuclear plant because it sits right on the canal near the Cape Fear River and resides within its estuary. And so because of this, we have to monitor aquatic organism populations and ensure that they're not being impacted as much. And so we mostly do this through two main samplings of impingement and entrainment. And so impingement, pictured there in the blue, is actually where the plant is taken in water via the pump systems. But before the pumps, we have these traveling screens that can actually carry the fish and other organisms to the fish return flume, which takes them back up that canal so that they can re-enter the estuary. But sometimes organisms are small enough that they get past these screens, such as larvae, and then they enter the plant and are discharged, which is what we refer to as entrainment, and that occurs at the green section right there. And so the overall objective with this data, it gives us a look at how energy production is affecting organisms, and also presents a unique aspect since this is one of the only power plants in North Carolina to sit on an estuary. And so it also helps us look at impacts from different life stages, because we do larval fish, but we also do juvenile impingement samples as well. But for this data, we were looking a lot at entrainment. And so we currently know a lot about what is in that intake canal, what's being entrained and everything like that, because we've recorded data since 1979, and then 5 years before, from 1979 to about 1983 was our baseline year. And then after that, we have further data. And so the most pertinent information to this is that we have, my letters or labels got messed up there, sorry about that, is that we have all that information on shrimp, swimming crabs, and Atlantic croaker, which were the 3 species looked at more closely. And Atlantic croaker is just a representative species for fish with this research. And so what we were looking at really was trying to see how effective these preventative measures at our impingement site is, and like how effective they are with preventing entrainment. And so the objective behind that, we analyze the number that are being entrained using the baseline and using a % reduction. And then also a statistical analysis to see if these 3 organisms have been treated equally in terms of the screen and preventing them from being entrained. And so from there, we kind of arbitrarily said, you know, if it's 75% reduction for entrainment, then our preventative measures are successful. And then if the % reduction rates per species for that same year is plus or minus 10%, we would assume that they're being treated relatively equally. It's anticipated that of the 40 years of data that we actually have that % reduction value for, the majority of those will be above 70% reduction, entrainment reduction. But we will also see natural influences from like hurricanes and flows. And then we also anticipate to see relatively minimal variability between species, just because ideally, we're not doing anything to target a specific species. So hopefully they're all being prevented from entering the plant the same. And so with our actual samples, this is done monthly currently. In prior years, it's been done like twice a month, weekly. So it's really died down, but currently we're doing it monthly. And for impingement sampling, imagine those bongo nets are separate. And so we drop them in the fish flume at our impingement site along the canal. And we leave them in there for about 2 minutes just because it collects a lot of stuff really fast. And then we pull them out, and there's a canister at the end of that net that we take back to the lab and empty into our gridded bacon pan so that we can start sorting out larval fish. And so we have a clicker and we count how many organisms we're finding. And then at the end of the process, they're identified to species. And then we record a minimum and maximum length. And so it's a very similar process with enchainment, but it's done at our discharge weir, also depicted in the map. And it's a very similar setup to this with 2 flow meters in each, or 1 flow meter in each net so that we can validate the flow. And then just a similar process with the observing them and finding out which species are in there. And so that was the field for the statistical analysis. I have our percent reduction rates over here. This one is pictured for entrainment of all organisms. This isn't also meant to be a result slide, just I used Excel to get this. Our percent reduction rate. And so also we have our number of entrained organisms, which we get from those counts that we have, and then also a expansion factor. And so just use that simple percent reduction rate for each species as well. And each species has a different baseline number. And then to analyze the difference between those 3 species, I used one-way ANOVA at first, but with some consultant, we observed that the variance was very high, which is a violation of ANOVA assumptions. So we tried one-way ANOVA by taking the log of the data, and then also the Kruskal-Wallis test, which does not share the assumption that variance has to be the same. And so the results from the percent reduction are kind of listed here. There were 40 years of data. Of those 40, only 10 of them were below an overall total percent reduction of 70. So these are you're looking at the worst years right here, unfortunately. But we can also see through these values our impacts from hurricanes. So that increases the flow, and then you're increasing how much water is getting pumped in to these cycles and just the chances of something getting entrained more. So yes. And then the statistical results, all of them reported a p-value below 0.05 and essentially 0. So we do see that there is a pretty big difference between those 3 species, which you might have even caught on this slide that Atlantic croaker is really being impacted still by entrainment. So that was very validating for sure. And then, so just discussion, pretty much everything that we've kind of touched on, that our preventative measures are overall effective because majority of them are 70% percent reduction from that baseline. And that baseline was the count of organisms entrained before the screens or the fish return flume were in place, which I forgot to mention. And Atlantic croaker seems to be impacted the most still. And then we can see that influence from the hurricanes. And then for the statistics side, they were below 0.05. So we see that they are not being prevented from entering the site equally. And then in the future, I would probably expand my data set. I was given yearly averages of entrainment, but to expand that to monthly, I think then I would maybe have a lot more flexibility with running some statistics tests. But yeah, I would like to thank Linda Hickok, who's my manager, Kyle Hussey, and he was my like main mentor, but also the entire water resources group does this each month, and it's a 24-hour shift, so day shift, night shift situation. And then Mallory Winters from our Department of Statistics helped me with that. And so now I'll open it up to questions with some honorable mentions of some species. Yes.
I think there's some sort of difference for the Atlantic croaker, like in the shape of the screens or the size of the
Yeah,
fish.
that is a great question. I kind of think, okay, that's actually the first time I've thought about it, if I'm being honest. It definitely could be just that they're more like slender this way, whereas shrimp, I'll go back a little bit actually. Yeah, they're a little like wonky to begin with, where they're just kind of like more at right angles and maybe get caught more. So I think it probably is just like the general shape of the croaker, probably that's doing that, because this mesh size is about our fine mesh. It's like super tiny. I think it's like 3/8 of a millimeter. So it's like pretty small stuff. But yeah, I think that would be my initial hypothesis. But yes. Yes.
There's seasonality to their spawning. I don't know what the right
There
term
is. Mm-hmm.
So do you think that the Atlantic croaker then they spawn during the hurricanes like season?
Yeah, it definitely. It could for sure. Normally we get shrimp and crab like yearly. So and that maybe takes away that aspect of seasonality a little bit for the croaker and their spawning. But they do tend to spawn. We see a lot of larval croaker over the summer, which is of course when they're being the hurricanes occur. So it could definitely be an impact for sure. Mhm. Yes.
What do you think you might see if you had that finer scale monthly data?
Well, really with that data, we change up our screen configurations, not often, but every few years, depending on what it is. So I would kind of hope to dive into those and see if those screen configurations actually have a significant difference. Because in the past, like the percent reduction table, the year before, or the screen configuration before 2000 was actually like 100% fine mesh, pretty much, which is our smallest grain and prevents the most. And none of those values are up here. But this is when we're trying to mix things up and introduce a larger mesh. So I would hope through that data, we could actually further evaluate and like see what's causing that impact and what would be the best, I guess, screen configuration to use out there. But overall, I feel like it would reflect the same in terms of the general species and how they're being treated. Yes.
Why is 70% reduction considered acceptable?
That was just kind of an arbitrary value that I was like, oh, I'm in college, so 70% is passing because and like for our requirement, we just have to have a reduction or we have to entrain less than our baseline, which we do quite easily. So it's like for the state standards, we're abiding by all of it. But just to kind of weed out things a little bit, that was kind of who's passing and who's not passing, pretty much. So, yeah. Yes.
Does the average flow matter at all?
So when we were initially going over the data, we were thinking that yes, it does. And we were thinking that more flow, more things getting trained. But when I started looking into it further, we didn't see as strong as a correlation as we initially would have thought. Our highest flow year, I think, was actually also one of the highest percent reduction years. So that was kind of an initial, that's not really following what we're thinking. And the flow would also kind of have to be dived into a little bit deeper because we were initially thinking more flow, more entrainment, but then you also have more flow, more potential for organisms to get caught in the sides of the estuary and the canal, and for them to actually be able to just escape and hang out in that intake canal without the potential of being entrained into the plant. So it can kind of go both ways with our ideas of what's happened. Yes.
And sort of a silly question.
Yeah.
Because this is all very foreign to me.
Yeah.
But could, speed of moving around or mobility be a factor here? Because you've got, I'm assuming the Atlantic croakers and teleos fish, right?
Yeah.
And then you've got two vertebrates that are presumably slow.
Yeah.
Less mobility and brawl. Is that a factor here?
I think kind of when they're this small and at this larval stage, for kind of a perspective, our entrainment organisms are about 5 millimeters large. So I think when they're kind of that small, they don't necessarily have much mobility at that point in time. But since you mentioned it as well, the reason why we actually look at swimming crabs specifically is because they can enter higher up into that water column and actually be taken up by the pumps. So a yes and no for your question. Yeah.
All right, thank you.
Awesome. Thanks, guys.
And finishing out our second session today, we have Julianna Welch.
Hello, my name is Julianna. Over the summer, I did a research survey on the vascular plant diversity of a meadow that was recently cleared of black locust in the Great Smoky Mountain National Park with a nonprofit called Discover Life in America. Oh, okay. Skip this one. Okay. So over the summer, I was an intern with Discover Life in America. They, like I said, are a nonprofit within the Great Smoky Mountains. Their goal is to connect communities and scientists, discovering, understanding, and conserving the natural world, which basically means they try to find every species in the Smoky so that they can conserve it, because you can't conserve what you don't know. But I worked with different park staff to conduct a research survey on a meadow right next to the office. It used to be agricultural land, and then it was, like I said, it had black walnuts, but they recently cleared it. So they were trying to see what's there now to figure out how to better manage it and just return it to a native meadow and native plants. So not much was known about what was growing there because it had just been cleared pretty recently. So I was just there to help bridge the knowledge gap and kind of understand what's there at that moment. And then just kind of help understand the area and the history of the land impacts and successional growth of the plants. So just a little bit of background on where this was. There's the Twin Creeks Meadow, which is in the Smokies. It's about 3 miles north of Gatlinburg, Tennessee, off of Cherokee Orchard Road. In the 1920s, it was purchased by a family, and they made their homestead on it. They had all kinds of structures. They had pools, all that. You can see that image. That's what it used to look like. And then it was donated to the Smokies. And while they were doing that, they introduced all kinds of species like Japanese maple, daylilies, things like that, which aren't traditionally found in the Smokies. And then when the park got hold of it, they turned it into or they removed the apple orchard that the family had placed on it, and then allowed black walnuts to take over. So another thing to note is black walnuts produce an allelopathic chemical, which kind of inhibits the growth of species in the area. So there wasn't much surrounding the meadow. There was poison ivy, blackberry rubus, things like that. So there wasn't too much else. And then just a little map of where I was right here where the star was, kind of near Gatlinburg, like I said. So in the park, that gives you a visual. So the objectives were just to determine the vascular plant diversity in the meadow. And like I said, it used to be an apple orchard and residential agriculture area. So they wanted to see how that impacted the successional growth in the Smokies, and just see how the history impacted it. And then just set up a baseline of data for the park. They are going to turn it into restore the meadow. So like I said, they were trying to find what was there so that they can restore it later. And then my prediction was that there are going to be a lot of early successional plants in the area. some of which were like the ones that were planted by the Voorhees family, like these lilies, which I found in the plot. And just like plants like giant ragweed, things like that, that are kind of typical with early successional, but yeah. So this is my methods. What I did is I had a 28 by 38 meter plot, and then I split it into 20 smaller 7 by 7 meter plots. So if you see in that picture there, I started with one here and 20, kind of snaked around, so it's just easier to walk in the area. And then each corner was like, there was a metal rebar that I like staked into the ground, and then it just had a tag. The numbers don't really matter that much, they're just kind of random, but it's so that the park can go back and create like a future similar study to figure out what's there after they conduct their management so that they can have future surveys. And then each plot was sampled in random order, and then the distinct morphospecies of the vascular plants were recorded. So in each plot, I would go around and figure out which species are there. I wouldn't record like the individual numbers of species, just like which ones are there. So it could be like multiple species in the whole plot, but I wouldn't count the number. And then species that I was able to identify in the field, I would record them right there. Just like the easier ones, such as like mayapple, stuff like that, stuff that I knew. And then anything that I didn't know, I would collect, and then I would like press for later identification. And then went back and swept each plot for a second time just to make sure that I didn't miss anything. And then every preserved specimen, I would go and like look through field guides, look at dichotomous keys, and then I also consulted with park ecologists to help determine the species, since they're more familiar, since they actually work there, with what's growing. And then if I couldn't identify a species or taxa to species, I would then try to identify it to genus. This usually occurred if like the fruiting bodies or like flowers weren't present just because of the time of the year, and we weren't able to identify it any further because you can't really identify a grass if there's no fruiting body on it. And then also qualitative differences when each plot were noted. So the relative sun exposure, which I use shade map to do, and then the overall biomass in each plot was also recorded. So for my results, I found 83 unique taxa in each plot. 71 were identified to species, 11 to genus, and then there was one that we couldn't identify. This plot right here is basically the same map as before, but it shows each number of species. So it ranged from like 55 to like 18. So that's just how many I found in each plot. There were 3 species that were found in every single plot. There were Japanese siltgrass, wood sorrel, and giant ragweed. And then there are 10 species that were found in only a single plot. So that was like trillium, mayapple, there was some strawberry bush, things like that. And then the species richness ranged from 18, which was over here in plot 17, like I said, to 55 in plot 3. So there was a pretty good like diversity in the small plot. This is like a building for scale, so it's not even really that big. So it kind of shows that there was just a lot growing there. And then this was like the map that I had created via shade map. You can see right here, kind of more toward the east, it was a lot more sunny. There was a lot more open area. And then here it was shady. This is likely due, there was one dead apple tree left over from when it was an apple orchard that was still standing. So that was just probably what that blue dot is, probably just an error in the program. But there are 43 families represented across all plots. So it ranged from like 12 to 29, just depending on where it was. And using the USDA plant database, I found that 12 species were either introduced or exotic. So that means about 14-ish, 14.5% of the species were introduced and were not native to the area. And areas in like the sunnier regions, they had a higher biomass. So there was a lot more like plant material there. Some of the plants got up to about 5 feet tall, just kind of based on like my height. So they were pretty big. It was kind of hard to walk through them in the sunny areas. I kind of had to like bushwhack. And then there was less plant diversity there. So there was a lot of like the same species. Like, for example, I've said like giant ragweed, that was all over these plots. But in the more shaded areas, there's a lot more diversity. So there's things like mayapple, trillium, there's some cancer root, but there was a lot less like density. So it's just really easy to walk through. Like those ones were easier to survey because I could just kind of look down and find them. So that was nice. And like I said, areas to the east had more sun, like you can see here, and areas to the west, they had tulip trees and then Chinese chestnut, so about 75% cover. So this is kind kind of like the results table. I did this for 83 of the, like all 83 of the species. So this is just like a little fragment. I couldn't include every single plant there. But you can see here, this is like the taxa, the family, and then I recorded like each plot that they were in. So like from 1 to 20, and then just if they were like in plot 7, I would just mark an X. And then I had commonness in each plot, so like how common each specific species was, and then the plot richness per plot. So like I said, I did this for 83 different plants. It's like a big long table in my paper, but that was like the chunk of like my research. So now for the discussion, there were multiple taxa that were present during the early successional stage. There's hemp dogbane, like I said, keep saying giant ragweed, there was a lot of that. They're pretty common with like the early successional stage after the clearing of black walnut. And then many plants that were not associated with early successional stage were over in the shaded areas because they'd been in the shade longer, and they didn't have as much competition. So that was pretty interesting to find out. No species were really surprising to find to me. Maybe the Japanese maple that had been planted by the Voorhees family was interesting, but we were able to figure out where that came from. There was a single like yellow white robin trillium that was found in a really sunny area. It was by itself. I don't really know how exactly it got right there. I almost missed it because it was like in between all kinds of other big plants. So finding that was kind of interesting, but I did find others in like the other side of the plot, but it was just kind of cool. Overall, I think it went pretty well. I think I could have gone like over the plants beforehand with someone who knew the area better, because I'm like from the area, but I didn't know every single plant. So just going and like trying to figure out everything was a little bit intimidating, but I got through it and I found it really fun. And then I wish that I'd taken a picture of each specimen that I didn't know before I preserved it, because the only unidentified species that we had was just because it didn't preserve well. Once we took it out to look at it, it was like, what is this? Like, it did not look the same. So if I had taken a picture of it, I might have been able to like figure out what it was. And then the baseline data will be used for the future. They have already started like managing it. This is a picture of like pretty soon after I completed the survey, they mowed it. There's already a deer there. I saw copperheads, turkeys, there was a bear scat, all kinds of stuff, so many insects. So they've already started restoring it. They planted milkweed, so they're getting going on starting to make it a new, like a meadow with native plants, which is really cool. So that was really fun. And then my acknowledgements, Will Kuhn, who was my mentor, I would like to thank him for sure. And then the park biologist that I consulted when I didn't actually know what the plants were, Jesse Webster, Troy Evans, and Ian Savo were great. And then other staff, Todd Butcher and Jamie Matzko. And then my other interns, Kareem Ortega, JC, and Pablo. So yeah, does anyone have any questions?
The east-west gradient associated with trees, did you notice any species identified with that east-west gradient?
Yeah, so they're like specifically like mayaples or like early successional plants. The way that I was able to determine the yellow wake robin was going through and looking at iNaturalist data in the region and like seeing what was there previously. So that was really helpful. So like the more early like spring plants, I saw there are small saplings, like there's red maple saplings, things like that. And then in like the way more sunny areas, it was just like, I'm not gonna say weeds, but just like the stuff that grows really fast. So there's hemp dogbane, there's clover, just things like that that you would find that tend to grow a lot faster, but then get outcompeted a little bit later when more plants kind of start going into the area.
I have a follow-up with that if anybody else has a question. So you just said iNaturalist, and that has me thinking, like, have you considered setting up a project for that site so that you could hopefully motivate visitors to log what, you know, because then you could crowdsource mapping succession. Right?
I think that would definitely be helpful. The Discover Life in America, where I interned for, they are really into iNaturalist. I have never used that app so much. It was so much fun, but setting up a project for that exact area, I hadn't thought about. It's pretty high traffic because there's like since there's so many buildings, there's like a little well house right above it that people would travel to called the Ferry House. So there are people like walking by all the time. When I was conducting my survey, I would just like be out in the field and there'd be people walking by. So it would definitely be useful to help involve the public. And then that is something to think about. I might-
Especially if your rebar markers are still there.
Yeah, they should.
Then you could even associate like what plot.
That's true. I should definitely like think about that because am still like, I'm hoping to still be associated with the Smoky. So being able to go back and help would be awesome. And then if I like have people doing it too and helping me, that would be great. So I have definitely something to think about.
First, how big did you say the smaller plots were?
They were 7x7, and then I don't remember the exact how big like each plot was off the top of my head. But each like one of these 7x7 or 28x35 meter was like the whole thing. So it was pretty big. That's one of the reasons why I didn't count each individual species, because that would just be so, or like each individual plant, I guess I should say. So that would be a whole bunch. So I just did it by species.
How did you like decide how big to make each of the plots? Just that we would be able to survey better?
It was just like the math that ended up happening. Since that's how big it was, I just kind of did some calculations and I'm like, what's the most effective way to like do this and kind of have like a nice number? So that's kind of how that fell. It was just
Is that 28x35 is the size of the meadow that is back?
yeah, that's the whole thing. So this whole thing is like 5x35.
I enjoyed your talk, Julianna. How are they planning to, are they planning to do any management to keep this open?
Yeah, so pretty soon after I finished the survey, they, like I said, they mowed it. They've been planting milkweed, or they did at like the end of the summer. Some of the other rangers were going out and like monitoring it. I wasn't as involved with the milkweed process, but they were doing that. I know that there are some talks of doing a burn there because it is a pretty small area, so it'd be pretty effective to burn it. And then I guess they're going to use the data too to just like see what's there and then determine the best ways to manage it based off of what's there. I won't be too involved in that process because that's more of like the resource management side of the park. But I know that, like I said, some controlled burns, prescribed burns, things like that, and then mowing, or what I know is happening now.
Are they planning to do any control of microspecium?
I hope so. There was a lot. Every single plot had microspecium. And then when they mowed it, you were still able to see like even easier how much was there because it's still pretty small. So maybe some like herbicide or something with that. I'm not too exactly sure about the management methods, but I would say so.
Back when it was an apple orchard, do you know if they totally just kind of like tilled up the soil or if they left any vegetation among the trees?
So this right here, this was my office. That's, or I think that is one of these buildings, but it was like, this is the exact same land. So it looks like it was pretty tilled, like just based on like the image. It was like right here, you can see some trees. So I'm pretty sure that they kept it pretty tilled, pretty like apple or orchardy. But yeah, there wasn't too much diversity there. They had planted some like daylilies and Japanese maple, stuff like that. So I think that was kind of how they kept it, was just like you would an old settlement place.
You said that they cut down the walnuts. What kind of legacy effects might you expect to see from the walnuts and be able to see legacy effects?
So because of the allelopathic chemical, during the time that they were present, there was not like really anything else. I should have included a picture of when they were actually walnuts in the area. I just didn't think to do that. But after, like, I visited in the spring before my internship and there was really like nothing there still. It had been roughly a year at that point. There was just like, it was like ankle high vegetation. So there wasn't much, and that was after like one growing season. And then this summer it just like exploded from when I visited, I think it was March, to when my internship started in June. There was like so much vegetation. It had just gone so high. So I would say for like the first year, there are definitely some like lingering effects from the allelopathic chemical, but I would say that those kind of subsided after the first year. I didn't do too much like study because of that, because I wasn't there last summer to do my internship. but just based on like what I saw, I would say that it kind of, after the first year, there wasn't as much present in the soil.
You said you had an unidentified species that didn't press well. What did you do with it? Did you pressing away or?
We ended up throwing all of the specimens away. The park didn't really have a use for them, and you're not really supposed to like keep the specimens from the park. I was only able to like gather them because I was under permit. Our thoughts were it was some kind of like ilex, like holly, but it just pressed so poorly. It was we didn't know what it was. That was definitely like something I should have thought of better. I've pressed plants before, but it's more of like a hobby type thing for me. So I wasn't like, I did what I knew. Maybe I could have like I said, taken pictures or kind of gone and checked them more often. But yeah, sadly I wasn't able to keep them. I kept like one single plant because I thought it was pretty and I was like, I can't do anymore. They had a good herbarium. The original thought was to put any like useful species in an herbarium, but there wasn't really anything, like I said, that was super surprising. And all the plants that I found, they already had records of, so they didn't really have a need for them.
Thanks, Julia.
Thank you.
We are here in our third installment of the Fall 2024 Applied Ecology Minors Research Symposium to celebrate 5 students' research. So I'm excited to learn from them today, and we will start with Kylan Rivera.
Hey, hi everyone, I'm Kylan Rivera. I'm a senior here at NC State, majoring in zoology and I'm minoring in applied ecology. For my applied ecology minor research project, I decided to do my project on different approaches of estimating carnivore diet. And yeah, before we get into all the different approaches and stuff, I just have a quick video to show you one way we can estimate what a carnivore is eating. As I see here, oh, hold on a second.
No, no worries, no worries. Go back here. And there we go.
Okay.
Does that work?
Yeah.
Right.
And
You can see it up here.
okay, yeah. Can I skip it a little bit? I was gonna skip it like halfway. Yeah, I'm sorry.
No, no worries. All right, where do you want to go?
Like a minute 30.
Here?
A little bit further. Yeah, like right there. Yeah. So yeah, this is one way we can estimate what a carnivore is eating. This is basically observational. As we see here, a lion they're attacking a zebra, and from this we can estimate that a zebra is a part of the lion's diet. So this is just one way, but I'm gonna tell you many other ways. And let me see. I'm sorry.
There.
Okay, thank you.
Now you are ready to go.
Thank you. Yep. So before we talk about the other methods, I just want to tell you why knowing what a carnivore eats is important. So knowing what is a part of a carnivore's diet is very beneficial and fundamental to ecology, as you can learn many things from an animal's diet. For example, you can learn where seeds are being dispersed. You can learn if there's any parasites in the area. You can learn if there's any predator-prey relationships from the diet, what the carnivore is eating. You can also see if there's any competition in the ecosystem too, if there's 2 animals eating the same vegetation or same prey. So yeah, this is just a couple things you can learn from what a carnivore is eating. Yeah, so there are 4 main methods of estimating carnivore diet. There's morphological, molecular, GBS cluster, and stable isotope. The main method is the morphological method. And this is basically just going through the scat and picking out bones, seeds, hair, and then from that you can tell what the animal is eating. And then another approach is the molecular. That is basically going through the scat and extracting DNA from the scat. And then from the DNA, you'll be able to see what the animal is eating. And then there's GPS cluster. This is from an animal wearing a GPS collar. And on your GPS map, you can see like different clusters where the animal has been like circling a lot. And that's usually like something the animal has killed or has been feeding on for a couple of days. Then the last method is the stable isotope method. And you can do this from extracting hair from an animal. And looking at the stable isotopes in the hair, you can see what the animal has eaten in that. Yeah, so what do we already know? We know how to quantify diet from scat, and we also know where to find scat. So we know how to see what an animal's eaten. And we also know that there are many studies that have showed comparison of different methods. There have been comparisons of morphological and molecular, or like morphological and GPS, but there hasn't been studies that have yet compared all 4 approaches. And that is one of our objectives for this project, is to quantify the differences between all 4 approaches. And our hypothesis for this was that there will be no differences between the approaches. And that leads to our alternate hypothesis being that there will be differences between the approaches. And our second objective was to find the useful sample rate from each approach. And the useful sample rate is basically saying out of all the samples collected, how many of those samples had data in them. So onto the methods. So the first thing we did is we did a literature search on Web of Science. And from this, we were looking for papers that compared at least 2 or more of the dietary approaches. And it had to be, the study had to be on an animal in the order Carnivora. And yeah, so that was what we were mainly looking for. And to do this, we used search terms such as carnivore, diet, comparison, and many other search terms to find all the papers. And once we had all our papers downloaded, we then went through them, and we were then moved on to the data collection part. And from each paper, we were looking for things such as the location of where the study was done, the animal the study was done on, how many samples were used, the diet, focal taxa, and many more things that we entered into our data set. Yeah, and so for the useful sample, for this, we just basically went through our data set and took the proportion of samples with data and then total samples. And we did that for comparisons between studies and then also comparisons between each approach. Yeah, so for our results, this is basically a map showing where we got all our data points from. So we had around 22 total different data points, and we got data from each continent except Antarctica. And we got a good variety of species too. We had lions, leopards, pumas, and many more. And each dot represents a different comparison. The reds representing morphological and molecular comparison, the blues representing morphological GPS, greens morphological stable isotope, and purples molecular stable isotope. Yeah, from this, we were able to see that we had 16 total different species from every continent, like I said. And we seen that our most common comparison was the morphological and molecular. We had 13 comparisons from those. And then our least common was the molecular and stable isotope with only 3. Yeah, so here's a graph showing morphological compared to every other approach. So we have morphological compared to molecular, the middle is morphological compared to GPS, and the last one's morphological compared to stable isotope. And yeah, so on the graph, there's a legend. The orange means that morphological detected that specific taxa better than the other approach. So for example, in this one, we see that morphological detected small mammals better than molecular. And then if it's blue, that means it overlaps 0, which means they're pretty much even at detecting that taxa. So for medium mammal, morphological and molecular were evenly detected the medium mammal. Yeah, so one of the main results from this is we've seen that morphological is better at detecting small mammals compared to the other ones. We've seen that between morphological GPS, and stable isotope, and morph and molecular, that morphological is better at detecting small mammals. Another thing we've seen was that morphological is better at detecting plants and compared to the others as well. And this is very important because a lot of the carnivores, a huge part of their diet is plants. So accurately estimating and detecting plants is very important too. And the last thing we've seen was that medium mammals, large mammals, and birds, they were all evenly detected between each approach. And this is our results for the useful sample. Some takeaways from this, we've seen that stable isotope had 100% useful sample rate, meaning that every sample from stable isotope had data in it. And we've seen that our lowest useful sample rate was the GPS cluster with around 30% useful sample rate. And we've seen that morphological and molecular had similar useful sample rates. Yeah, so one of our main takeaways from this is that the morphological is probably the most efficient from this, because we've seen morphological is better at detecting specific taxes such as small mammals and plants compared to the other ones. And also morphological has a high useful sample rate too, so you're getting a lot of data when you use the morphological method. Another thing, like I said earlier, that GPS had the lowest useful sample rate, and when you're doing GPS cluster, you get a lot of samples, but you see it only had a little bit of samples with data. So that's something to take into account when you're doing this. You just know, like, if you do GPS, you're not getting as much samples or having a high sample rate. And another thing was that stable isotope had 100% useful sample rate. So the takeaway from this is that maybe using stable isotope, if you want to be 100% sure that you're accurately getting samples with data, maybe stable isotope may be the best approach. But keep in mind, we did have low number of samples for this. Yeah, we faced a few challenges doing this research project. One was that we didn't get an even amount of comparisons for each approach. Like, we had a lot of morph and molecular, but we didn't have like an even amount for like morph and stable isotope, molecular stable isotope, or molecular and GPS. So we didn't have an even amount of comparisons for each. So that was one challenge we faced. And another one was that we had small sample sizes too. We've seen, I think we had 22 total papers, and like for stable isotope, there was only like 22 samples. So we did have small sample size, but we still got some good data. And another challenge, our last challenge, was that there may have been a taxonomic bias, meaning that we had in the order carnivora, we had a lot of carnivores that were the larger carnivores, such as lions, bears, wolves. And the challenge is this: we didn't get many small carnivores, such as like raccoons and skunks. So there may have been a bias in our results because we didn't have full range of all the order carnivora. But we were thinking to overcome this, we could maybe ask researchers who have done comparisons. We could ask them, but they haven't quantified the data. We could ask them for their data, and then we can quantify it ourselves and then add it to the data set. Or we can just conduct a study on our own and make sure, like we can choose what carnivores to do and make sure we get even amount of approaches used. Yeah, so how can we apply this? We found that most of these methods complement each other, meaning there was not one that was clearly better than the other. So they would seem like some was better at detecting small mammals. GPS cluster was good at detecting large mammals. So we've seen that, and papers suggest that maybe using more than one approach may be the best thing to do, because you're getting the best of both worlds, and you're getting the most accurate results too if you use two or more approaches. Yeah, I like to thank my mentor, Dr. Alex Jensen, in the back. He's been such a good help for this project and contributor. Really like to thank him, and also Dr. Mariel Rodriguez Carras. He's been a great help too. And any questions? Yeah.
Detection of the small mammals with the morphological approach. Like, what do you think is better about that approach for small mammals?
Yeah, I think that maybe it's the bones. Like, you can maybe calculate the bones more, and like maybe like if it's molecular, like maybe the DNA may have been washed by rain or something if you're doing DNA. But if you're just going through the scat, you might be finding the hair or the bones in it. Compared to the other ones are like they're mainly like testing. And for GPS cluster, I guess it might kind of be hard because they might eat the whole thing too. But yeah, you're welcome.
So if you could set up like your perfect dream experiment where you're collecting all of those data from the carnivores, would you focus on a specific like bears or big cats or wolves, or would you focus on a specific taxonomy of carnivores, or would you try to sample across all those different types?
I'd say probably across different types, because there's so many different ones. Like, I guess a bear would be a good one because you're getting like they kind of eat, they're like omnivores. But maybe if you do like a raccoon or a skunk, their diet's way different than a lion. So I'd say probably a wide variety, just to get like make sure to get the whole order of carnivore just in general. So yeah, probably a variety.
As long as you're designing the dream, just go really big.
Yeah, probably, yeah. Lions, skunks, bears, as many animals as possible.
Thank you so much, Kylan.
Thank you.
Up next, we have Andrew Orovic.
Hello, everybody. My name is Andrew Orovic. I am a zoology major here at State, triple minoring in horticulture, business administration, and applied ecology. And today I'm going to take you guys on my adventure to find bats. Welcome to Wildwood Park. Wildwood Park is a 229-acre wetland with a 90-acre lake in the center. It is just on the outskirts of Harrisburg, Pennsylvania, my hometown. And it assists the public by reducing flooding, improving water quality, and supporting biodiversity. While Wildwood does have a purpose as a nature preserve, it focuses heavily on educating the public. This past summer, I had the chance to intern here at Wildwood, where my boss gave me the opportunity to research anything I wanted, anything of my choice, and I chose to do bats due to some prior interest. In Pennsylvania, there are 9 species of bats, and they all belong to the Vespertilionidae family, which basically just means that they're insectivores, they eat insects. They play an important role as predators, a host for parasites and pollinators. And according to the Pennsylvania Game Commission, the PGC, they have an importance as a bioindicator. As insectivores, bats are sensitive to things like chemicals like insecticides, and a change in their abundance may reflect changes in populations of arthropods. Economically, bats help reduce the cost of pesticides and food crop production. In the United States, the estimated reduced cost of pesticides was around 3.7 to 53 billion US dollars due to bat predation, which is quite a bit of money. While bats don't have like an issue with predation, they do have predators, but it's not really a big problem. They were seeing like declines in their populations. The cause of this is us, unfortunately. Wind turbines, highway mortalities, introduced diseases, and hibernation disturbances are all factors that go into their reduction in populations. In particular, wind turbines are, I think it's an average of 50 bats can die to one wind turbine in 1 year. To put that into perspective, Pennsylvania has 27 wind farms. A wind farm has on average 50 windmills each. So 27 wind farms, 50 windmills each, 50 bats per windmill. That's about 68,000 bats per year just in Pennsylvania. But what's even more crazy is that's not the biggest problem. The biggest threat to bat populations in Pennsylvania is white nose syndrome. White nose syndrome is a an exotic fungus that causes fungal infections in bats. It was caused by cavers, unfortunately, not cleaning their equipment and going cave to cave. And it has caused an overall decline of 99% of all migrating bats in Pennsylvania. 6 of the 9 species in Pennsylvania have been affected by white nose syndrome, 2 of which are now endangered, the long-eared bat and the Indiana bat, and 1 is being proposed as endangered, the tricolored bat. Now, Wildwood has no prior research on bats, which is one of the reasons I wanted to do my research on bats. I think that Wildwood can provide habitat that nurtures bat populations. Just down the road from Wildwood is the Susquehanna River. The Susquehanna River has a pretty big bat fly problem. And it's so bad that for the past 23 seasons, they've had to spray weekly to keep these populations at bay. Now, what if Wildwood could be a place that nurtures bats in a way that these bats can help us manage these bat fly populations? Well, I wanted to see if bats, if that's even probable. So my goal was to set the groundwork for further research. That is right in the way. I didn't even notice it. I had nothing to compare my research to. Unfortunately, like I said, Wildwood didn't have anything. So my goal was to provide that for other people. Maybe my research can be used to help other people continue what I want to do. Before I did that, what just happened? Before I did that, oh, what happened?
It migrated. There you go.
Interesting.
Click.
That's good.
Okay. You just need your cursor.
All right.
Find it. Before you did that.
Yes. Okay. I had to answer a few questions. My main question was, are there bats in Wildwood? I don't even know. There had been sightings, but there wasn't any solid evidence. So I needed to go find that out. And once I found that out, I had some secondary questions. What species are specific to Wildwood? Where are they roosting? Where are they eating? What can Wildwood do to help improve these bats' habitats? And what programs can they run to better inform the public? So to start, I did a risk assessment. I knew that I was going to be bringing people along with me to do these surveys. It was going to be at nighttime. So I needed people to know what they were getting into. So I performed a risk assessment, and then I had to become more familiar with bats in general. I had no idea anything about bats. So I made a sheet of every bat species in Pennsylvania that had behavioral and feeding strategies that each bat has, and their characteristics of each. I also watched several videos of different surveys that have been done in Pennsylvania to see how they were doing it. And I took notes of areas around the park that may be good areas for bats to roost or feed. I found 6 different places around the park, and they were split into 3 ways: the top, the middle, and the south of the park. The park is very elongated, as you can see here. I was looking for areas that had like open water or a high insect density. And that's kind of how I chose the areas. And from there, I had to choose a date. I chose between July 30th and August 16th from 8 p.m. to 10 p.m. According to the PGC, bats around this time were beginning to disperse. The pups had learned to fly a few weeks earlier, and now they're beginning to leave their roosts and go to other roosts. I thought this would be a good time for me to go and see exactly what the park has. So I gathered my materials, binoculars, watch, and everything else you see up there. And I had to plan how I was going to take my notes. So I took note of bats by the minute. So for 1 minute, I'd count what was in the air, and then I'd go into the next minute, count what was there. And I was doing this so I could see the activity levels throughout the night. I also noted any defining characteristics I saw of each bat, like a white stomach, or maybe they're quite larger than the others. In addition to that, I took note of other factors, like the temperature of the day, the weather, if there was something going on that was causing a lot of traffic or loud noises, stuff like that. And here are my results. I don't know if you can see it. That little guy right there is a bat. There's bats in Wildwood. And no, not just that one bat. In fact, we counted a total of 303 bats over the course of the survey. 5 different species were somewhat identified. We weren't able to just say, this is it. But these were our best guesses. We had the little brown, big brown, tricolor, red, and silver hair. On average, the bats were most active from 8:40 to 9:10. My father, who was with me the whole time, actually would look at his watch and be like, okay, it's 8:40, and look up, and we'd be seeing them right away. It was kind of funny. And in the middle of the park, we saw the most activity. On the bottom, kind of average, and on the top was the least. And what's interesting about this is that when we were in the middle portion, we noted that every bat came north to south. So we expected to see bats in the north, but as you can tell, there wasn't really many. So to recap, do bats live in Wildwood? Yes, they do. 5 species, in fact. Like I said, the little brown, big brown, tricolor, red, and silver-haired. They're most spotted along the towpath, which is the left side of the park, and the morning glory, which is right at the bottom. And they're most active 30 minutes after sunset, from 8:40 to 9:10. Now, we did run into some challenges. Like I said, there was difficulty identifying them. As you can probably tell, at nighttime, every bat kind of looks the same, and a big white spotlight isn't going to help you much. But we did our best with the behavioral sheets we had and different characteristics that we were noticing. And we did have some trouble finding good viewing places. Being unexperienced in this field, it was kind of difficult to determine where we should go, but we did make changes on the fly. For example, the meadow was an area that didn't have any water at all, and we didn't see anything while we were there. So we took a short walk and we saw stuff. So for our next survey, we decided to go there. And that's how we kind of made changes on the fly. We were also unable to find emergence points. This was to be expected. With 6 surveys, you're not really going to find exactly where they were, but we did our best. And I think with further surveying, we would be able to find these emergence points. There were also some inconsistent and uncontrollable factors. For example, there was a large event that happened directly next to the park during one of my surveys and caused this huge influx in cars. I couldn't control that, but it happened. So what can Wildwood do? Wildwood already does a lot of this, a lot of habitat management, for example. But there are some things they could pay more attention to, like keeping snags or making more brush piles. They already avoid chemicals in their water, and I know that they've been restoring their wetland for quite some time now. This is a long-term goal, and luckily, a lot of these help a lot of the environment, not just bats. So it's a good thing. They can also continue surveying. In Pennsylvania, there is a volunteer program called the Appalachian Bat Count. It's basically 2 surveys throughout the year, one in the beginning, one at the end, and you give in your results to the Game Commission, and they monitor the health of different bat populations around Pennsylvania. I think that's one way Wildwood can help out. And I think they can also do programs. Like I said, Wildwood is very focused on their education stuff. So maybe you can teach people to make bat boxes. Lead some guided walks about them towards nighttime. It's not super late. And maybe we can make it a topic for field trips. Teach children young that bats aren't scary, but they're our friends. And also, I'd like to highlight that Wildwood can make Wildwood-specific handouts. They do have PA basic handouts, but I think it's more personal when it comes from the place you're in. I'd like to thank you all for listening to me, and I also give a special thanks to Chris Rebert, my mentor, who gave me this opportunity and led me along the way. The Wildwood staff, who were constantly there, whether they knew what I was doing or not, they constantly gave help. And my father, who came with me on every single survey, whether it was raining, the mosquitoes were biting like crazy, he was there. So thank you all. Questions?
You have a comment that it seems like your sampling might be really well suited if a master's student was to consider this either, NC State or from Penn State. I know they've got a big life sciences school up there in State College, Pennsylvania, that you could consider some type of occupancy modeling, so you could quantify this with some relatively basic statistical models. And that would be really informative to Wildwood Park, because if they're going to manage their habitat to try to get bats in the park, they're going to want to see some type of bang for the buck. And occupancy modeling using data either collected now or next summer over the next couple summers, and then comparing it down the road after some habitat modifications are done, that could be a really slick tool that the managers of the park would want to see it, so they can evaluate whether their habitat mitigation programs are achieving their intended results.
Yeah, okay.
That's a lot of little brown bats, especially like this day and age. And so I know you didn't do like building surveys, but I'm wondering like if there could be a maternity colony nearby. So do you know if there are any like old barns or like buildings in the area where those bats like could be hanging out in the summer, or nothing like that?
Well, there are areas. Actually, there's a lot of like mines and tunnels nearby. I think that they could be. I think the summer roost is probably the most like. I didn't mention this in my I should have mentioned it. I didn't. But a lot of bats are very partial to different trees. And I actually looked at an old tree survey while I was in Wildwood, and found that they have almost all of the trees that these bats like. The American beech, the American ash, and a few others. And during the summer, they'll go in these roosts in these trees because they have trouble regulating their own body heat, and those trees help them regulate their heat. So I think it's more likely that they're roosting somewhere in the trees than a barn or something like that. In Pennsylvania, the most common bat is a little brown, so I wasn't super surprised with those results, but yeah.
Is Wildwood open at night?
Wildwood closes, I believe, at 9. I think it's around there. I can't remember. I might be completely wrong. It might be like 8. But they have done programs in the past that have gone into the night. For example, they've done owl programs, and they have attempted to do a bat program. It fell through for some reason. I can't remember what they told me, but they would open it to the park like for that reason.
I mean, now you can.
Yeah.
Right? That's awesome, because a lot of parks close at sunset.
Yeah.
So your window would be, yeah, that's great. Other questions? All right, thanks, Amy.
Up next, we have Christian Choto.
Good afternoon. My name is Christian Choto, and I did my research in the Sustainable Health Ecology Lab at NC State that primarily researches snakes. I did my study in snake teeth, which is a surprisingly understudied area. Let's take a look at how diet is related to teeth. Herbivores primarily have flat teeth, which is useful for grinding plants, and carnivores have teeth for tearing meat, and omnivores like humans have a contrast of both. However, mammals are not the only ones that have dental variation. We all know a little bit about snakes, that they have fangs. The fangs are primarily used for venom delivery. These fangs are hollow and specialized for venom delivery, helping subdue prey before swallowing them whole. Venomous snakes also have other teeth besides fangs, and these fangs are less known about. Most North Carolina snakes are colubrid. I hypothesize that teeth may vary between colubrid species based on their dietary preferences, like mammals. Mammal-eating snakes restrict prey, while amphibian-eating snakes tend to go after less dangerous prey. These different strategies led me to believe that their teeth differences may be correlated to diet. I examined 41 specimens representing 7 snake species, 2 of which were mammal-eating snakes and the rest were amphibian-eating snakes. For our specimen collection, most of our snakes came from the NC Museum of Natural Sciences, and when we got them, they were from roadkill, so most of them were in rough shape. Due to this, I chose to do the snake length instead of counting the snake teeth. Unlike humans that have 1 set or row of teeth per jaw, snakes have 4 on the top and 2 on the bottom, as you can see in the picture on the left. For this study, I extracted a tooth from each row, from the bottom, top, middle, and top outer. I found some complications when taking them out due to how small they could be and fragile. In instances where I was unable to take the tooth out, I extracted the entirety of the row, as seen in the top middle. After extraction, I was able to place the tooth underneath a microscope at 40 times magnification. After this, I used an ocular micrometer to measure the angle at the longest axis. Snake size affects tooth size. For instance, on the left, that is a garter snake. His teeth are barely visible due to its size. Then we have the python. It is larger and its teeth are quite visible. To control for this, we measured snout vent length, SVL. Starts from the head all the way down to the cloaca, using a string to measure. Next, I'll show you 3 graphs, bottom, top, middle, and top outer, representing the jaws. For the y-axis, we did the tooth length, and for the x, we did the snout vent length, SVL. The red points will be for amphibian-eating snakes, and the blue will be for mammal-eating snakes. Tooth length did increase with SVL. SVL always had a significant p-value. And contrary to my hypothesis, there was no significant difference between the amphibian-eating snakes and the mammal-eating snakes. The R-squared value, which measures how much variation in tooth length, is explained by SVL were high. The top outer graph did have an outlier, which caused it to be an R-squared value of 0.5, much lower than the other graphs. I'm showing the same graph again, but the colors are now for snake species. Take note of the green highlighted by the green arrow. This is the hognose snake. I was only able to dissect one, but once dissecting, I found out that it had a specialized fang. And this fang is primarily used for its prey of choice being toads, and it is a toad specialist. The fang allows it to pop the toad and deflate it for consumption. In conclusion, snake size is proportional to teeth length. A 3-foot amphibian eating snake will have averaged the same tooth length as a 3-foot mammal eating snake. However, the average mammal eating snake tends to be larger, thus having larger teeth when compared to the average size of an amphibian eating snake. The hognose snake was my only major outlier due to its rear fang, the rear fang being specialized for its prey of choice. For future research, instead of taking the fang for the hognose, I would like to take the non-fang parts of it for my data. along with other dietary choices through other snakes, like the queen snake and the water snake. And aspects for tooth morphology, I could check and see for curvature and pointiness. I want to thank the NC Museum of Natural Sciences and Jeff Bain and the Sustainable Health Ecology Lab for their contributions to my research. Thank you for your attention. Any questions?
The admission, I know nothing about these topics so far. Would there be a difference if I think all those snakes were non-venomous, right?
Correct.
Would there be a difference in any of these relationships? Do you think if some of these snakes that you researched, possess venom instead? I mean, would there be some different relationships compared to what you found?
The only one that I did study that had venom would be the hognose, but its venom isn't for humans. It's primarily for amphibians. So other than that, I'm not sure, but maybe.
So you kind of lead into my next point that I wonder if you're onto something in researching potentially in the next cut of this, researching the venomous snakes for the non-venomous snakes and leaving the hognose out of the non-venomous group because you had a lump together in your graphing and then consider the venom versus non-venom as an additional factor in your future XY relationships for plotting purposes and see if there's some differences there between the venomous and non-venomous groups.
It could be. Mostly with the non-fangs would probably have correlation.
Yep.
Yeah. Don't we clamp them, right? Clamp.
Don't have any contact with the venomous snakes. They're dead, by the way.
Up next, we have Sean Sullivan.
Good evening, everyone. Thank you for coming. I am Sean Sullivan, and I'll be talking about the work I did on the recreational angler perceptions of where to vent demersal reef fishes with barotrauma. Just click here. Start with my acknowledgements today. I'd like to acknowledge everybody listed on this slide for their work in assisting our project. I'd also like to acknowledge, of course, my mentor, Paul Rudershausen, my co-authors, Jeffrey Buckle and Brendan Rundy. I'll go into some pertinent background information, set the context for this research. Barotrauma, you could call it pressure trauma, is just what happens to a fish when it's brought up from significant depth. The internal gas in their body expands. This causes damage to their internal organs. It's mainly a problem in phycoclystic fishes, and it's a significant contribution to discard mortality, which is a problem in fisheries management because if you have a significant portion of your released fish dying after you release them, the effectiveness of size limits, bag limits is eroded. You can see in these pictures some of the classic symptoms of barotrauma. Up in the top corner here, this fish is its stomach coming out through its mouth. It's filled with air, eyes popping out of its head due to all that internal pressure. In this bottom picture here, you see a fish trapped at the surface because it's essentially a balloon now. It's too positively buoyant to resubmerge, swim back to depth. Of course, this has been a recognized issue for some time. So mitigation strategies to reduce that discard mortality have been developed. The subject of my work was venting, which is a very, very simple procedure. It's basically popping that fish, take a hollow needle, insert it into the swim bladder, and you let that trapped gas inside escape, allowing it to become less positively buoyant, ideally letting it swim back down to depth, capture, taking away some of that stress involved with swimming against all that air trapped in its body. There is some hesitation to condone this method, though, because despite the fact it's been proven to be effective, its effectiveness has only been studied when it's performed by trained researchers. All the people doing this research are fisheries biologists, and well, we know it's done when it's done the same way again and again, it's effective, but there's no information to get at the effectiveness when it's done by people who might not be well-trained like the anglers interacting with this fishery. And the information on how anglers vent and where anglers vent is mostly unknown. That's why we wanted to do this research, was to collect information that would basically inform future work to determine what discard mortality percentage is associated with improper or unconventional venting techniques. The objectives of our study specifically were to determine relative angler familiarity in the U.S. South Atlantic, which is our fisheries management region, and to determine some of those anatomical locations that anglers are selecting to vent fish, relate that to their experience with the technique and maybe their experience fishing aboard a private vessel. To do this, we surveyed anglers. We ran our survey for about a year, taking trips aboard the boat you see here, which is a head boat. It's a charter boat, essentially. Takes anglers offshore, about 50 to 60 people at a time. We go with them, we interview them, and this boat primarily targets snapper grouper species. So the fish that are suffering barotrauma means these anglers are engaging directly with the fishery where barotrauma is most prevalent. Our survey process was extremely simple. It consisted of one original question, which you see here, which is, have you ever heard of venting? We asked this to every participant. If they said no, they had not, that was the survey. Mark that down and move on. If they said yes, they had heard of venting, we gave them this sheet here. Asked them a couple more questions. Have you ever vented a fish? Gets at their venting experience. They fish off of a private vessel as opposed to a charter vessel with any consistency. Should add that question is only asked with a subset of anglers who were given this sheet here. And then the crux of our survey, we prompted them to mark a sketch on this fish to basically tell us where they would vent a fish if they were to do it. Once we had a big stack of paper surveys in hand with a bunch of little X's and circles and checks on them, we wanted to delineate the regions that we could sort the marks into. Outlined in black is the traditional region. It's the location where it's generally accepted correct to vent a fish. That's what we call the proper location. Most literature would support that as being the right place to vent a fish where the swim bladder is under normal conditions. That dashed line is the non-traditional region, likely effective based on some published works to reduce discard mortality, but less studied. And then outside of that is the incorrect region. This is where there's no information to support the effectiveness, but you could reasonably assume that venting a fish here is not going to let gas out, or it could further injure the animal. We did some statistics, chi-squared test, to determine the difference in regional selection between the experience groups we delineated with the yes or no questions. The results of our angler familiarity questions, we talked to 567 people on that boat in 22 trips, and less than half of them actually had ever heard of venting. So only that 232 got the paper survey where they were allowed to answer the rest of the questions. 37% of those 232 had vented before, and 102 people were asked that question on their private angling experience. About half of them did in fact fish aboard a private vessel in their own time. Now in this this figure, you see all 232 marks split between those who had yes vented a fish before and those who had no not vented a fish before. Black is a mark in the traditional region, blue is a mark in the non-traditional region, and red is a mark outside of the traditional region. You can kind of see where on the fish's body some of these people are choosing to mark. You see the clusters, the cluster there around the anus, a cluster on the averted stomach. And you can see that the images are very similar. It's because there is no statistically significant difference in the regions marked by either experience group. Pretty similar whether or not they've vented a fish before. This is the 102 marks given by the people who had experience or didn't have, sorry, let me backtrack. These are the marks from the people who actually got asked that question, the subset of that 232. Again, no statistically significant difference based on the response to that question. So we can consider everybody essentially in the same response group. And you see here how it breaks down amongst all surveys, roughly 60% are in that traditional, non-traditional area, and about remaining 40-some% are in the incorrect area, somewhere outside the delineated regions. Those suggest to us that there is, in fact, widespread unfamiliarity with venting among recreational anglers in the South Atlantic. Additionally, a large percentage of anglers we surveyed were identifying potentially harmful or ineffective locations to vent these fish. This confirms to an extent hesitation by fishery managers to condone venting as a barotrauma mitigation technique, but I think it highlights a need for further research into where exactly, or not where exactly, but the mortality associated with venting fish in these improper areas. And that, again, like I mentioned before, was part of what motivated us to do this study, and hopefully what this will carry on into. With that, I'll take any questions.
Be the most effective way to educate anglers? Like, is there a particular
Yeah.
time, place, or format?
So I should have touched on that. There's actually very little educational material out there. I think the most effective way would be to hand out instructional pamphlets with a license, or maybe make it so people need to be exposed to instructional material before they can buy a license to fish in ocean waters, or at least fish on snapper grouper fishes that suffer barotrauma. But anything is better than nothing, which is what exists now, at least in the South Atlantic. The Gulf Coast has done a lot better with trying to educate anglers, but it seems like that might not even be a great way to increase their familiarity with the technique. So.
I know this was like a formal survey question, but I assume like you also just like chatted with a lot of the anglers about venting. Did you find out like where do they learn about it? Like when they know about venting, did anyone tell you like?
Usually it's because they've seen somebody else do it. They're out on their friend's boat, they see their friend do it, or they're out on these charter boats and they see basically a professional do it. Somebody who works in the fishing industry as a part of the fleet. So. Yeah.
Another related question. With the venting tool itself, when you buy that, assuming buying like Amazon or something, does the packaging tell you where you should do it?
Most people don't buy them. And no, it doesn't, because there is no like 100% approved location. There's a lot of untested locations, and there's the venting location along the side of the fish where it's known to be effective, where a large body of research has proved that it is effective at reducing discard mortality, but that's not included with packaging or anything you would get if you bought a product. So. Yeah.
Your anglers like use the other terms besides venting for your process?
There are other terms. Venting is definitely the most contemporary, most widely used. Fizzing is another one. I think when it was first invented 20-some years ago, fizzing was pretty popular, but if they don't know it by venting, they wouldn't know it by anything else. So. Yeah, that's all. Yeah.
Ashlyn, you wanna come close us out?
Hi, I'm Ashlyn Walter, and today I'll be presenting my work on mycelial growth of ectomycorrhizal in sufficient and insufficient phosphorus conditions. Just to introduce you to this topic, ectomycorrhizae is found in the rhizosphere in soil, and it has associations with many plant species. In this case, we'll be looking at loblolly pine. And these associations are beneficial to plants because they connect through the roots and increase root surface area, which is important in nutrient acquisition for the plants. Many micronutrients and macronutrients in the soils are not always in an available form to plants, and these mycorrhizal associations help the plants obtain these nutrients in a form that they can use. For loblolly pine, This is a very commercially important tree to North Carolina and the entire southeastern United States. These plants are in sylviculture. We produce a lot of wood here in North Carolina. So there is a need and a reason to invest in the care and the upbringing of these trees. And so when you're fertilizing trees, when you notice that there is a phosphorus deficiency, for instance, NPK fertilizers will be applied, but not always is this completely necessary. Phosphorus is a very immobile macronutrient. It doesn't leach easily from the soil, and it's not always in an available form. Depending on the soil that you have, such as Ulta soils, which are the native soils that pine trees mainly grow on here in North Carolina, the acidity of the soil tends to bind very tightly to phosphorus, and it's very hard for plants themselves to chemically break those bonds, which is where ectomycorrhizal communities come into play, because they essentially bridge that gap and help the plants out by breaking those bonds with enzymes. And so that brings me to the objectives of this project, which is to identify ectomycorrhizal species for the purposes of developing communities which will be responsive in low phosphorus conditions. We want to see if there are any trade-offs between ectomycorrhizae when grown in these conditions, because there might be fast-growing species that achieve far hyphal growth, but they might not have the same biomass accumulated if they were to be grown slower. And we just wanted to see how different species react in these conditions. This is all for the purpose of curating species for a synthetic ectomycorrhizal community. Sorry, I wasn't actually on the objective slide. But the prospective species that we gathered data on, there are many species in literature that have been documented for decades associated with loblolly pine. But many of those species aren't featured today because the data took a little too long to accumulate. You can't make these species grow any faster than they do. But we do have Physolithus tinctorius, Rhizopogon fuscarubens, and Acephala macrosclerotiorum. And we'll get into how we collected data. So for the methods for this project, it's fairly straightforward. Since we're looking at hyphal growth, we'll be growing the species on plates in low phosphorus, sufficient phosphorus conditions, as well as just regular MMN medium. And when we're taking our subcultures of the isolates were grown on MMN, and we're taking them from the subcultures onto new plates with a 1 centimeter plug, and we're putting them on the plates and putting them in an incubator to leave them to grow. They're grown in the dark at 29 degrees Celsius, and for each species, there's 10 plates per treatment. And then also alongside this, we're taking measurements whenever there's a sufficient amount of mycelial growth visible. And this is just macroscopically with the naked eye. And the mycelial growth will help us, we'll get more into the measurements of that. But in ImageJ, we wanted to try out something new. Previously, it was all done by hand, which the other undergrads informed me was very painstaking. So this was a new method that we're trying out, trying to get very accurate area measurements through setting a scale in ImageJ of the image, and then just going through manually making polygon lines along the Sharpie lines that I took day by day. And for the biomass, these were conducted in liquid medium, just in low phosphorus and sufficient phosphorus conditions. The same thing, we took the thallus and we placed it in the liquid medium and left it in the incubator. But we leave these in there untouched for about a month. However, some species, when we went to harvest, we noticed did not have hardly any significant biomass growth. And so we made the executive decision that many other species needed to just be left in the incubator for maybe 2 or 3 months of growth. Because once we got done drying in the oven, there would have been hardly anything left. There wouldn't be anything to measure. And when we're harvesting, we're taking out the thallus from the medium and we're bathing it to rinse off the medium, and then we're putting it onto paper towels and taking a scalpel to remove carefully the hyphal growth from the agar plug itself. And we're weighing the fresh material, then drying it in the oven for 5 days at 70 degrees Celsius, and then taking the dry mass as well. And just to backtrack a little bit to the hyphal growth on the plates, I just wanted to showcase what that looks like. This is tinctorius on the MMN media. And as you can see, I just wanted to note the growth is very homogeneous, like radial growth is circular. And these plates grew really fast actually compared to some of the other species. This was done growing at 31 days. Some species are still growing in the incubator right now at 70, 80, 90 days. And on sufficient and low phosphorus conditions, the presentation is a little bit different. It took around the same amount of days for the growth to be completed as well, just around 33 days. And the growth is a little bit more irregular, and a little bit more filamentous. And this is a little bit more easily visible, like when you're taking the measurements, it's less dense, and this is just due to the conditions that it's grown in. This is just a preliminary graph. I just wanted to disclose, I don't understand what was wrong with data set 5 in R. I couldn't figure out what was wrong with it, but they all stopped growing. We stopped taking measurements at day 33. So just ignore that part of it. But all of the growth was relatively similar. They start off growing basically at the same rate from 5 to 10 days, 5 to 12 days. And there's a little bit of variation, but not much. And then they all taper off around the 33-day mark. And then preliminary results for the biomass revealed some interesting relationships between the species. As you can see, Porifusca rubens was not very significant in biomass growth. And we saw the most biomass growth in the tinctorius species. And further statistical analysis is done in R to determine if the biomass accumulated in low phosphorus conditions and sufficient phosphorus conditions is enough to be significant. And running an LSD test, we determined that the biomass achieved in all of the species across species interspecifically, they are significant. But there is no interspecific significance, meaning that in tinctorius, for example, the medians are different between low phosphorus and sufficient phosphorus, but essentially they grow the same under both conditions, which when I get into discussions, this is promising information. And like I mentioned before, Fusca rubens is very insignificant in biomass. And Acephala macroscleratorium performed not as well as tinctorius did, but is still showing no significant difference between low phosphorus and sufficient phosphorus. So what this means is that tinctorius is a really good candidate for the ectomycorrhizal community. This will be seen in sapling inoculations, which I'll talk about more in a minute. But since there's no significant difference between those low and sufficient phosphorus conditions, we can expect to see that transfer over to the saplings when inoculated. The saplings should be able to grow in these low phosphorus and sufficient phosphorus conditions and produce biomass that is significantly statistically the same as if it were in either condition.
And in the other two species, they did not achieve, well, Tinctorius was the only species that was able to achieve full hyphal colonization of the plates, meaning that we don't have enough data to conduct our t-tests to look at significant differences between the growth speeds of the species. So the next steps would be, as you can see in the top right, those are all of the plates that have still yet to achieve full colonization. And there's more of the biomass jars on the bottom that still need to be harvested. So there's much more work to do. It's just on the time scale of these species themselves. I already mentioned the t-tests, but I'll move into sapling inoculations. We want to observe any effects and any potential benefits that these species have on the sapling growths. And from this experiment, I hypothesize that when inoculated with the Piscithylus tinctorius, the saplings will show no statistically different growth rates and biomass accumulated between either treatments. And more steps down the line, this will help develop the synthetic ectomycorrhizal community that's consistent of multiple species, and this will benefit agroecosystems that are suffering from poor soil conditions such as low phosphorus. And I wanted to thank my mentor, Maria. She was great, and everyone at Garcia Labs. Any questions?
Yeah, really nice talk and a really heady topic, so congratulations on a nice project.
Thank you.
What's the cost? Like, I live down the coast of North Carolina and there's a lot of warehouser lands that grow yellow pines for wood fiber production. What would the cost be for those kind of entities to consider this type of inoculation program to grow yellow pine? Is it cost feasible, or?
I actually do not know the back end costs of those. I do know that all of our materials are very expensive. So conducting research like this in general is just time consuming and expensive due to the time consumption it takes. I don't think the saplings themselves are really like the tree and the material themselves are the cost issue though. But I am not very versed on the back end costs of everything.
Victoria's achieved like total plate coverage.
Mhm.
And it looks like in your growth graph, like notwithstanding that wild point, everything seemed to plateau by 33 days.
Mhm.
Do you think those growth patterns would change if you gave it like a bigger petri dish?
I do think so. I think that the species, since it's native to North Carolina environments, it is trying to grow farther. This species in literature is shown to increase the general biomass of the loblolly pine trees. So this is a very strong candidate, and I think some of the plateau for other species as well, not just Tinctorius, some of the species that have stopped growing or slowed basically to a halt that I couldn't include in the data for this presentation, I think it's due to the constrictions of the plate, but I can't be fully positive on that. I guess I don't have a statistical analysis to prove that, but that's my hypothesis.
Is it possible, like, have access to bigger plates?
We do have bigger plates.
What do you want, but to spend like 3 more months? waiting for those to
Exactly,
grow?
exactly. But I think that would actually be worth it. I've seen it been used, just we didn't use it for this project specifically.
It'd be cool if you could push out that.
Mm-hmm. I agree.
Well, thank you so much, Ashlyn.
And that concludes part 3 of our fall 2024 symposia. So thank you all for coming out. Happy last day of classes and have a great break.